Welcome, Mr. Saurabh Sengupta, and welcome, Dr. Shaud Lahi.
Thank you. Thanks.
Thanks. Hi, everyone.
হ্যালো হ্যালো হ্যাঁ অভিষেক শোনা যাচ্ছে আমার কথা? আচ্ছা ঠিক আছে ওকে ওকে।
Ujjal Dev, Mr. Ujjal Dev, are you here? Sir, can you start? Okay, sir. Thank you, sir. Welcome you all on today's webinar Spectrum of Advanced Biological Science that jointly organized by IQAC, St. Paul's Cathedral Mission College and the Department of Microbiology. If you have any questions during the presentation, please type them into the message box. We'll bring them up during the presentation, and we will also have time for questions at the end of each session. Now, without further ado, I'll turn the time over to Sudhanuka, faculty of our microbiology department. Over to Sudhanuka.
A very good evening to everyone present here. I take this privilege to warmly welcome all our delegates to the international webinar jointly organized by Department of Microbiology with IQAC cell of St. Paul's Cathedral Mission College on Spectrum of Advanced Biological Sciences. We have 4 eminent speakers in today's session who are now working in US, Bangladesh, Canada. But before starting the main session, according to the traditional ritual of our college, we must praise the Lord through our prayer. For prayer, I would like to invite Mr. Untitled David, coordinator of our college sector.
Okay, good evening, everyone, and I hope I'm audible to everyone. This is such a great opportunity, and this is such a great initiative. Let us pray even as we start. We believe that we need God's blessings even as we start this evening. Shall we pray? Our Heavenly Father, who is the creator of heaven and earth, Father, we thank you that it is you who sustain, it is you who help us invent, it is you who help us create. Thank you that you are a God who is interested in us and interested in progress and science and all the knowledge that exuberate, Lord. Heavenly Father, we especially pray for this evening. Thank you that as St. Paul's Cathedral Mission College, from the microbiology department and different partners, we could come together and host this international webinar. We thank you for the opportunity. We thank you in times like this. We can come together on a platform like this. We can discuss on mental health. We can discuss on different issues pertaining to us. And I believe that speakers and the receivers, each of us will be tremendously blessed. We pray for every digital platforms here. We pray for all the networks, internet facilities, and different social network platforms. We keep everything under your care and protection, that each one of us will be able to have uninterrupted internet source, will be having uninterrupted audio network issues. We'll be having a time where we're able to enjoy and receive from the great resource person who are there this evening. We also pray, Lord, this evening, especially for our resource person. Thank you for their lives. Thank you for the way you have brought them this far. Thank you for their success. Thank you that they are the best in these fields. As we hear from them, thank you for their expertise. Thank you for helping them to invest in the life of St. Paul's College and all across the world. We pray that you will strengthen them, help us to understand them, what they're going to present, and help us to also take away and to look at progress in the coming days. We also pray for the microbiology department, also for our TIC sir, and for each one of us in the panel from our college. We pray that each one of us will be able to align and innovate and cooperate and look at progress, not only for our college, but our country, India, and all over the world. We thank you, Lord. We keep everything under your care, your protection over all of us. In Jesus' name we pray. Amen. Amen. Thank you so much. Have a blessed time.
Amen.
Thank you so much, Mr. Dev. We have our TIC of our college, Professor Devashish Mondal, with us. Now, I would like to request her to share a few lines of theme for today's symposium.
Good evening, everybody. Thank you, Sutanuka Basu, professor of our department in microbiology. It's my pleasure to announce that for the first time, the Department of Microbiology, which started its journey in the year 2001 and introduced honors in 2003, now is arranging an international webinar of this stature. And the topic is spectrum of advanced biology. And it is doing it, this webinar in collaboration with the IQAC of the St. Paul's College. I'd like to convey my heartfelt gratitude to the different parts of the world and who have accepted our invitation to shed light on different aspects of biology. I would like to convey my regards and thanks to Dr. J.R.P. Alfred, Secretary of Governing Body, our IQAC coordinator, Dr. Subhasachi Mahapatru, for their constant support, and also Professor Abhishek Das, convener for arranging this seminar, and all the teachers of the Department of Microbiology, as well as all the participants and the students, and also our Untitled David, who has started this seminar with a prayer. I am sure that all the participants will be really richly enriched by this webinar, and also we will know much about different aspects of biology. May the good God bless you all, and I wish the success about this seminar. Thank you very much, Shutanuka.
Thank you, sir, for your valuable speech. We have with us Bursar of our college, Professor Shubhendu Kanjilal. I would like to request sir to share his view on this seminar.
Hello, am I audible?
Yes, sir.
Okay.
Yes, sir.
Okay. Respected teacher in charge, St. Paul's Cathedral Mission College, all distinguished guests and dignitaries from different parts of the globe, my esteemed colleagues, and finally my beloved students. We are all assembled here in this webinar on spectrum of advances in biological sciences to learn about different microbiological phenomena, its application, and obviously a thrust must be given on the prevailing pandemic of COVID-19. All the invited speakers need no introduction for their commanding works in their respective fields. It is indeed an honor on my part to welcome you all to this college, which started its odyssey on 24th January 1865 and has earned a heritage of 156 years. In the long checkered history of this college, down a whole century and little more than half, the college passes through many occasions of highest elation and deepest despair, victory and vanquishment, and in this dynamic fusion of polarities are sown the seeds of the dreams. The basic aim of the college is to impart education of sound learning, building of characters, and to orient and sustain the students in search of values. In conformity with the spirit of this college, the different departments of this college arrange seminars, workshops every year in order to keep the students and the faculty members updated with the modern advents of the subject. But this year, the situation is quite different. The entire globe is now shivering in fear of coronavirus disease 2019 caused by RNA virus SARS-CoV-2. Officially, the disease was first reported in one Jinyantan hospital, one city, Hubei province of China, in the middle of December. Since its outbreak in the middle of December 2019, the disease has now spread over 216 countries and territories over the entire globe, affecting 3,26,73,432 people, with a death toll of 9,90,576 as on 27/9/2020 at 11 a.m. CEST. India is now the second worst affected victim of this pandemic, where already 59,92,532 people has been infected, with a death toll of 94,503 persons as on 27/9/2020 at 8 a.m. as per report of Ministry of Health and Welfare, Government of India. Considering the country-wise infection, India is at the first position for the last 2 months. The disease basically has thrown a challenge to entire civilization. Amidst this unprecedented crisis, the Department of Microbiology of this college, in collaboration with Internal Quality Assurance Cell of the college, has organized this international webinar through electronic mode instead of our conventional face-to-face seminar. I must take this opportunity to thank all the faculty members of the Department of Microbiology, Physiology, and Zoology of our college, and also the IQAC coordinator, Dr. Shobboshachi Mahapatra, for arranging this webinar. I must thank Mr. Ujjal Dev, to grace this occasion. He is also an alumnus of this very old college. In fact, no word of appreciation is sufficient to justify the untiring effort and dedication of the faculty members of the Department of Microbiology for making this webinar a reality. Basically, the students are also facing a typical crisis at this moment. I must mention here that the long history of human civilization tells us that we have every reason to be hopeful, because disease outbreaks often ravage humanity. So let us be hopeful. My heart goes out with this batch of students of second, fourth, and final semesters who have to face many challenges of this pandemic, which we are still trying to understand and grapple with. I personally believe that the students who are facing this pandemic and facing new challenges will come out more resilient, more powerful, and capable of handling different situations. These students will be remembered by history not for what they have lost, but for the facts, the change that they will bring in due course due to this pandemic. Today, we shall learn from the distinguished speakers about the several applications of microbiology in biological modeling and also some issues related to COVID-19. Hope that all of us will be immensely benefited by attending this webinar. Finally, I thank the organizer for organizing such a time-relevant webinar and also for giving me an opportunity to say a few words. Thank you all.
Thank you, sir, for your enriching words. We have with us our IQAC coordinator, Dr. Shobushachi Mohapatra. I request sir to say few words on today's seminar.
Am I audible? Hello, am I audible?
Yes, yes, yes.
Yes, sir.
Good evening, everybody. Respected teacher in charge, Basha, all distinguished guests and dignitaries from different corners of the globe, my colleagues and my dear students. On behalf of the Internal Quality Assurance Cell of St. Paul's Cathedral Mission College, I welcome everybody in this international webinar entitled Spectrum of Advanced Biological Sciences, organized by the Department of Microbiology and IQAC of this college. Biological science is the study of living things and their vital processes. The field deals with all the physico-chemical aspects of life. The modern tendency towards cross-disciplinary research and the unification of scientific knowledge and investigation from different fields has resulted in significant overlap of the field of biology with other scientific disciplines. We are fortunate to have Mr. Saurabh Sengupta, CEO of Ilomo, USA, Dr. Muhammad A. Rahman, Professor of Biochemistry and Molecular Biology from the University of Chittagong, Bangladesh, Dr. Indrajit David from the Department of Biophysics, University of Michigan, USA, and Dr. Sagar Lahiri from the Department of Chemical Engineering and Applied Chemistry, University of Toronto, Canada, as our distinguished speakers in this webinar in spite of their busy schedule. Their valuable lectures will help to pursue the higher education and research in biological sciences. I wish the webinar to be a grand success. Thank you all.
Thank you, sir. Dr. J.R.B. Alfred, are you here?
Yes, I'm here.
Oh, sir. We have the privilege to have you with us in today's session. We have our college secretary, Dr. J.R.B. Alfred, with us. I request sir to speak few of his valuable words on this occasion. Please, sir.
Good evening to all of you from this part of the world, and good morning to the others who are joining us from the other side of the world. I am so happy that the Department of Microbiology and IQAC of St. Paul's Cathedral Mission College, Kolkata, are hosting an international webinar on spectrum of advanced biological sciences. I, therefore, would like to wish and express my deep sense of gratitude to Dr. Abhishek Das, Department of Microbiology and convener of this webinar, who along with his colleagues from the Microbiology Department has taken this initiative. Thanks also go to the teacher in charge, Dr. Debashish Mandal, our Vice Principal, Dr. Gautam Banerjee, and our bursar, Dr. Subhendu Kanjilal, as well as to Dr. Savyachi Mahapatrak, IQAC coordinator, for giving all the support to see that this international webinar reaches out to the widest audience possible. And especially to Dr. Ujjwal Dev today for his initiation prayer. It is a delight to see that this is an international trust webinar in the truest sense by the various topics that will be presented by such distinguished speakers who are not only from all over the world, but as you can see, also from some of the best institutions worldwide. We are therefore ever so grateful to Mr. Saurav Sengupta from USA, Dr. Indrajit David, also from USA, Dr. Mohammad Atiyar Rahman from Bangladesh, and Dr. Sagar Lahiri from Canada to accept to participate in this international webinar and share their knowledge with all of us here. It is interesting to see that each speaker will highlight on topics of both academic and social relevance. The topic on student mental health in next decade, for example, is an important as we are aware that all students suffer emotionally. And it is known that around 40 to 50% of the student population experience high stress that manifests into cognitive, behavioral, emotional, social, and physical challenges. This talk will probably help us and the students how to achieve, maintain, and enhance their emotional well-being. The next topic is also of interest, accelerating rare dissociative processes and biomolecules using selectively scaled Md simulations. I probably would not be very sure, but I think it's based on the published work on molecular dynamics of simulations. This can be a powerful tool for modeling complex processes such as ligand unbinding, though many biological relevant dissociate processes occur on timescales that far exceed the timescales of typical Md simulations. We therefore look forward to hear from the speaker of how they are implementing and applying an enhanced sampling method in which specific energy terms in potential energy function are selectively scaled to accelerate dissociate events during simulations. The next topic, sirs, is the mechanistic outreach of functional foods to defend respiratory virus. A very interesting topic, probably. Will give an insight into the novel and indigenous sources of phytomedicinal components as alternative therapeutics for non-communicable chronic diseases, especially type 2 diabetes, it seems, and chemical-induced acute hepatic toxicity through the intervention in animal models, but primarily based on phytopharmacological and biological effects, and more so of Bangladeshi medicinal plants and plant parts, which are yet unveiled and which are being mechanistically justified. We hope to look forward to this, sir. And the final topic, how to get into a scientific career and how to be successful in it. Wow, that's great. And what I learned from my journey so far. Certainly, it looks like an autobiographical speech. And I'm sure this will create an interest in young researchers hearing from an experienced senior researcher with a demonstrated history of working in the biotechnology industry where his skills have been in protein purification, molecular biology, biotechnology, drug development, and hardware diagnostics. Wow, these are the 4 wonderful and important topics we are looking forward to. And we are really looking forward to hearing all this from these distinguished invited speakers. Thank you, especially to the college and to all the others for giving me this opportunity to be part of this wonderful and useful international seminar. And I'm sure that all of you who have joined in are looking forward to it as much as I do. Best wishes and congratulations to all who have put this together. Thank you. And let's sit back and listen to these wonderful lectures. Thank you once again.
Thank you so much, sir, for your enlightening words. I would like to thank once again all our previous speakers. Now, the much awaited moment has finally arrived. Now, and we are going to start the main session of today's international webinar on spectrum of advanced biological sciences. Our first speaker is Mr. Saurabh Sengupta. He is CEO and co-founder at ILUMU, New York, USA. Mr. Sengupta is an entrepreneur and intrapreneur with proven track record of building analytical solutions for consumers and businesses. Expert in launching novel tech products, solution, raising funds, managing milestones, building teams, and acquiring early customers. He is going to speak on the topic student mental health in the next decade, which is very relevant with this pandemic scenario. Over to Mr. Sengupta.
Thank you. Thank you, everybody. It's a pleasure, an honor to be here and present the case that is so dear to my heart. And I'm really honored that I will be talking to you over the next 15-20 minutes. And I would be happy to answer all the questions that you would have. So why don't I get started? Share my screen. And if you want me to stop and explain something, I'll definitely take a pause and answer your questions. So feel free to raise your hand and ask any question that you may have. Do you see my screen? I have just loaded the presentation with ILUMU name on it. Can you guys hear me and see my screen?
Yes, sir, we can.
Cool, great, all right. No conversation these days, at least in the last 6 months, can start without talking about Corona a little bit. So I would start there and then get into the right topic that I'm here to talk about. Often we think of Corona and look at the number of deaths by country and try to gauge who is better off, who is worse off. I wanted to mention that Corona has so much universal effect on all of us. Maybe death is not the right lens or not the complete lens. We should look at Corona from an economic perspective. We should look at Corona from a mental health perspective. The tail of this virus is going to be anywhere between 4 to 5 years, where so many different mental health issues that has come up as a manifestation of this virus, that I personally feel death is not really the right lens. A country can do much worse, especially a country like India, where a significant amount of the population is not is struggling to meet their daily wages. It's a much more challenging thing for them that may not be captured properly by capturing the number of deaths. So I just wanted to say that in my presentation today, where I'm going to talk about student mental health, I have particularly kept out Corona out of the equation. The reason why I have consciously kept out Corona out of the equation is because the student stress due to Corona has not worsened that much. It might come across as counterintuitive to you, but from the research that we have done, the student mental health situation was already so much worse before Corona that there is not much opportunity to make it even worse than what it is. 50% of the student population is stressed, severely stressed, due to many different things that are happening in their life. Corona might have impacted that and made it from 50 to 55. But even before Corona, about 50% of the student population is dealing with severe stressors in their life. It is affecting their mental health, it is affecting their physical health, and it is affecting their ability to learn and take proper decision that would create an accomplished career in future. That's why it's such an important topic today, because mental health and academics are not really isolated events. If somebody needs to learn properly, they need to first be able to be in a position where their mental health is allowing them to learn and grow to be the person that they want to be. So that's just my initial summary. I wanted to set the context as I walk through the presentation. You would see that I'm not talking much about Corona, and it is because Corona has only marginally impacted the student stress situation. It's already much worse before Corona. I have been in an entrepreneurial and startup ecosystem for the past 4 years. Before that, I used to work mostly for consulting companies, Deloitte, KPMG, and other consulting companies. But for the past 4 years or so, I'm in the startup and entrepreneurial ecosystem. I have built this company to reimagine student well-being. I don't think much is happening on this spectrum, even though there is a lot of talk around mental health, but truly solving the problem from the student's perspective has not really yet gathered momentum, which should. We are already quite delayed. That has manifested into 50% of the population being severely stressed. So we should do something about it. And with that purpose in mind, I have built Illumo. Our objective is, in the next 3 years, we want to bring the number down to 20%. If we can accomplish decrease of 30% of people suffering from severe life stressor, 30% of students experiencing severe life stressor, we would think that the objective for which the company is built is accomplished. I'm predominantly focusing on high school and college students because they are the most severely stressed part of the population. Elementary and middle school is still not yet as badly hit as these two populations are. And generally elementary and in elementary school, or as we call primary school, there's a lot of involvement from the parents and teachers due to the ratio of student teacher. Generally there's a lot of intervention that the students get, so it never becomes unbearable for students in primary schools. But when it comes to high school, as the student teacher ratio goes down, the students are developing their characters and they're interacting with other students who are also developing their characters. That socio-economic emotional interaction makes things very hard for high school students and college students. Especially for college students, as they are nearing graduation, the worry about their future career starts to make situation much more overwhelming, especially in an economy where there is not tremendous amount of work happening right after graduation. So that's one of the major reasons why high school and college students are so much severely stressed. We are going to look at many of the reasons why they are stressed. We are going to look at what are this stressors manifesting into, and how can we resolve it? We are going to look at all of that over the next 20 minutes or so. This is, I'm going to talk about some of the major stressors that we have found out. We have done a survey of about 600 students focusing on high school and college students. And we have found out that almost 71% of the students identified academics as a major source of stress. When I say major source of stress, I need to define this term. It means that somebody is feeling severely stressed at least a couple of times every week, where they feel that their feelings, emotions are not in their controls, their behaviors are not in their control. That's the definition of severe stress, where they are experiencing it at least a couple of times every week. And almost 71% of the students indicated that academics has been one of the major sources of stress in their lives. 43% of the students said family is a major source of their stress. Now, from family perspective, there are wide variety of reasons that can be captured as family. It can be parental expectation. Sometimes parents expect too much of their kids. It can be disengagement of the parents. While at one spectrum we have parents who want so many things from their kids, there is another tale where parents are not involved at all. They don't care about what their kids are doing. And that is also immensely stressful, because the kids feel that they are alone in this journey and they have nobody to share their struggles with. Apart from that, conflict at domestic situations, especially between parents, are a tremendous amount of stress source. Loss of family members, whether it's parents, whether it's grandparents, whether it's siblings. So loss due to death is a huge stress that every student go through at least 1 or 2 times in their student life. So that's a major source of stress. Social expectations happen to be an extremely major source of stress. It is a time when they are trying to learn the social nuances, what is fun, what is not fun. So all of this become extremely complicated for students to deal with in the early high schools and even early colleges, especially in India, where there is this difference between co-education versus gender-focused education. It becomes very complex for students to deal with, especially if they're coming from a boys' school or a girls' school, and now they're going into a college that is co-ed. It's a very different social dynamics that they struggle with. So that's definitely a big part of the equation. Friendship. When I was doing this research and I found out that friendship is a major source of stress, I found it quite counterintuitive. I always thought that when you are dealing with stress, your friends and family would basically steer you away from the stresses that you are dealing with. Yet 25% of the students identified as friendship to be a major source of stress, especially because peer pressure is part of friendship, right? You can't really isolate peer pressure and friendship completely and say these two are completely separate. So it kind of boils down into a situation where your friendship is stressing students a lot. Relationships are also a big part of this entire dynamics. Your friends having relationships and you not having relationships can create a tremendous amount of stress, because we often develop a sense of self by looking at what's happening in the life of our friends. That's how we try and understand if we are on the right track. That's kind of how human nature is. We can't really think of ourselves in isolation. Our self-beliefs, our self-understandings are always mirrored among the friend circle that we belong in. So when something happens in our friend's life, especially something that is beneficial that are happening in our friend's life and it's not happening to us, we stop and ponder if there is something wrong with us. So that's a major source of stress for students. And obviously, about 1/4 of all students find career uncertainty, job prospects a major source of stress. These are just some of the major source of stress. There are many others, but I only wanted to point out the top 5 causes that are stressing our students in the last 3 years, I would say. Where is the stress leading them? There are so many different repercussions or manifestations that happen due to the severe stressors that students experience. Depression is one of the major repercussions. I want to make a distinction here between depression that is clinical, which is categorized as a mental illness, and depression that is developed due to a stressor in life. Those two are not the same. Clinical depression is something that somebody has. It's like migraine. There is nothing you can do about it but to treat it, right? It's internal, it's inherent in us. That's clinical depression. But not necessarily when I say depression here, I am referring to clinical depression. I'm referring to depression that we all go through because there's a situation that we are dealing with. So I want to make that distinction that it is not mental illness. It is rather a manifestation of a life stressor that impacts somebody's mental health. And students go through depression a lot. There is nothing wrong about it. It is nothing related to their inherent things that they are dealing with. It is the life that is making them depressed because they are not able to find their way out of it. Addiction is another coping mechanism that students use when life gets overwhelming. One of the ways when they don't know how to deal with it, they look for alcohol or many other addiction. For some people, gaming is actually an addiction, right? So there are many different forms of addiction that students find just to cope with the overwhelm that they experience due to the stressors that are happening in their lives. There are some correlations that we can draw among socioeconomic state versus how the depression will manifest, whether a student belongs to a high socioeconomic family versus low socioeconomic family. All of this kind of correlates with the manifestations of the stress. But these are generally where we have seen that across the board, no matter what socioeconomic background you come from, depression and addiction, lack of self-esteem, sleep deprivation are all part of that general universal manifestations of stress. Loneliness is something that is really one of the major manifestations of stress. Not many people talk about it, especially from an Indian standpoint, where we are habituated to deal with so many people every day. It may not feel that a big amount of students are lonely, but that is the case. Even if somebody is interacting with their friends and family quite often, and friends and family have no idea that this person internally feels very lonely. And that is one of the major reasons that leads to suicidal instances. So, there is a high correlation between loneliness and suicidal tendencies, which is very difficult from a peer perspective or a family perspective to pick on, given that it is very intimate. Loneliness is a very intimate experience that student go through. Indecision is another major thing that students experience, given that there are so many things that they are trying to do and life has not yet prepared them well to optimize or decide how to make optimized decisions. They are often confused and they tend to take either wrong decisions or be indecisive, where they are not able to take any decisions at all. These are all, many different manifestations. It's kind of a little jumbled up here. Depression and addiction, depression and loneliness is more on the emotional spectrum. Indecision is more on the cognitive spectrum. So there are many different spectrums, but I wanted to highlight 6 most noted or identified manifestations of stress, and wanted to bring it to you, that if any of you are experiencing any of these, it is only normal. As I said, about 50% of all students are experiencing this. Many people may not be talking about this, but about 50% of the people are experiencing this. So if you have 4 friends, law of probability says that 2 of them are definitely showing some amount of stress that either you are aware of or you are not aware of. So, be mindful of these statistics, as it is done on a significant sample size. It's done 600 high school and college students. Now I want to make a point about, because it is about student life, the primary objective that we all have from students is learning, right? But then we need to stop back and ask ourselves this question: can a brain in pain really learn well, right? Our prefrontal cortex kind of shuts down when we are experiencing severe stress, and if we are experiencing this twice every week, you can imagine how much disruption it can cause in our lives, right? In a student's life. And as teachers, as academicians, as people who are dealing with students day in and day out, we need to be mindful that we cannot really expect a lot of learning from the students if their brain is not in the right place. So that's the major thing that we need to be mindful of, that it's not about academics. You can't really make somebody learn anything in isolation of what's happening in their life. Our brain can't really process learning, memory, or any new information if our brain is not in the right place. So unless we solve the biggest problem of how to deal with stress, how to reduce stress, it is probably not going to be optimal to think that somebody would be able to go through learning without really showing us that they are not able to do it appropriately. I want to talk about how the landscape is changing. It's very important for us to understand, and when we devise a solution of how to deal with student stress, we can't really devise a solution if we don't know about the future. What kind of problems are going to be faced by recent high school students when they will graduate, or maybe the current undergraduate students when they will graduate and they are somewhere 5 to 10 years down the line in their respective careers. If the future around us is changing, we need to be cognizant about that fact, and that creates a lot of friction in our life, because we were probably not cognizant of how the future is shaping up around us. So I wanted to put this together. These are the 6 major trends that are going to make our life severely stressful, or it can open up a ton of opportunity from a student perspective to design your future and to make sure you are reaching for the right path when you're deciding for your careers. There is a major shift in demographic landscape that is happening across the world, India being one of the major driver in urbanization, where we see a huge population influx is moving from rural areas to urban areas. And that creates a ton of problem in itself. There is a study that has been done on the correlation between stress and noise level that in your physical surroundings, and it has seen that noise generally stresses us up, and in urban environment the noise level is always much higher than what you will find in a rural environment. That is just one example how urban life is more stressful than a rural life. But yet, because of the opportunities, because of the ambitions we have, a big population, a big part of the population is going to live in urban areas than rural areas, and that are going to definitely create a tremendous amount of stress, whether it's noise, whether it's traffic, whether it's the lack of proper design or development of a city where it may not be able to hold the amount of people that live in it. If we look at some of the examples like New York, Tokyo, in India, if we look at Mumbai, New Delhi, we can understand how a city is not yet designed for so many people to live in, yet they are living in those cities. And that has its own set of stress that are very specific to the residents of those cities that other people may not even understand, right? But we need to be cognizant about urbanization when we are thinking about the future career or future mental health of ourselves. We should make conscious decisions rather than subconscious decisions that can make our life much more stressful than we would ideally like to be. Automation is another important trend that we observe today, where the workforce needed to get a task done is reducing every day because of the implementation of automation. If we look at the car industry from 1960s to the car industry today, and if we think about car industry another 15 years down the line where there will be autonomous cars, at least in some part of the world, you can imagine how automation is reducing the requirement of human labor. So we need to design, again, our career, our aspirations, rethink what are we studying, what are we not studying, so that we can be in line with what the future holds. We don't want to study something that would not be helpful for us because the landscape is changing so much. And we need to understand that also creates a lot of anxiety around students because they are unsure what they are studying today. Can it be implemented 15 years down the line, right? You don't know that. The entire landscape, especially on technology, is changing so fast that how can you be sure that what you are studying today is relevant for the future that we are going to experience? So I always think, learning happens in two parts. One part of learning is learning the subject that you are learning, and another part of learning is learning how to learn, so that you understand that it is a continuous process. Just because you have learned something that has been rendered not important doesn't mean that you are not important. If you have acquired the knowledge of how to learn, you can always learn new thing and make yourself or adapt to the future that will unfold. It's very difficult to predict the future. It is easier to adapt to the future that we would experience in 10, 15 years down the line. But at least we need to be mindful as we are studying. We need to be mindful how the landscape is changing around us so that we make conscious choices. Collaboration is another very important thing where we usually meant collaboration is a human-to-human collaboration. But now that landscape is changing quite a bit, where it can be machine-human collaboration that has become very, very important these days. And there are already applications that we see. If you use smartphones, and there are obviously voice-enabled technology like Siri in iPhone, Alexa for Amazon, where you can interact with another machine and get the knowledge that you need. So human-machine collaboration is becoming very, very important in days to come. Aging population is something that we are all going to see. The entire world. India is a little better off that way, but the entire world is aging right now, and overall that is going to throw a lot of challenge our ways. Given that the population is aging, the retirement ages are going to be longer. There was a time when people used to retire at 55, 60. Now in US, the general retirement age has become 70. It might increase further to maybe 75. And when these things happen, it becomes difficult for finding proper employability, because there is continuance of employment from the aging population. Additionally, that creates a lot of burden financially. Living is not free; it's expensive, especially when you have an aging population. Their medical needs are a lot more than a younger population, so that creates additional burden on the economy. So we need to be mindful of this trend to understand and design our future around how to take decisions, or how to develop a mindset that when we experience this, it is not really coming to us as a surprise. We are already aware of the future to a certain extent, how it will unfold. Connectedness has generally been seen as a positive thing, but with increased connectedness, there is always lack of privacy. So we need to understand the pros and cons of connectedness. Not necessarily connectedness mean that everything is positive. It has its negative connotations. One specific example will be social media, where we connect with others. It feels great to be connected over social media, but social media stress is real. People are projecting their lives as if it is their life, only the positive things, without really projecting anything that is happening negatively in their life. And we, as the receiver of information, we assume that that's the yardstick through which you should measure life, and that creates a misleading tone for what life should be. And when we can't live up to that standard, we feel stressed, we feel depressed. So connectedness obviously comes with its own negative stressors, and we should be aware of these trends. Climate change is something we should all be we should all be worried about. We should all be actively participating, because climate plays a huge impact on our mental health. I'm not going to talk about climate change in general and how it is important for our survival as a race. That's not the agenda of this conversation. But climate in general has a tremendous amount of correlation with how we feel, how we behave. If I can take an example, if you look at the Scandinavian countries, where it's most of the time it's categorized as one of the happy places on earth. If you think of Denmark, Sweden, they are mostly categorized as one of the happiest places on earth. But you look at their climate, and you look at the suicidal rates there, you can't really make sense of it. What's happening? Why the suicidal rates are so high in countries that are categorized as happy? And then you look at the sunlight, the amount of sunlight they get every year, and suddenly it makes a lot of sense how our weather and climate plays such an important role in how we feel, how we process our emotions. So it is something that we should be mindful of and take conscious decisions, right? When you are, I'll give another maybe practical example. A friend of mine was in India, and he was great. He was a great computer scientist, and he got a job in Sweden. He loved it. He said, this is the first time he'll go out of country. He will live in Sweden, and he's from India. He's from Calcutta. He's getting sunlight 280 days a year, and now he's going to Sweden, where he would get sunlight for like 50 days a year. It's very different lifestyle, and it stressed the hell out of him. And he decided that no, I cannot live in Sweden sustainably for a longer time. And after a year back, he moved to US. Now he lives in California, and he gets like 240 hours, 40 days of sunlight. But that's how our mind and body is built. If you need what you need from a nature and climate perspective, you need to be consciously taking career choices. Nothing is an isolated decision for you to live a life full of mental well-being. So what can we do? If our objective is to learn better, have an accomplished life, have an academic career that helps us develop, go for our aspirations, I think the first thing we should be aware of is how to develop better mental health. We need to make sure that our brain is right so that we can process information, we can have better memory, we can learn new things quickly, so that we can reach for the stars that we are meant to reach for. I want to now talk about the landscape a little bit. There are companies that are reimagining student mental health. We are obviously one of them. But I want to also talk about two other competitors that I have immense respect for. You at College is an app. They are offering a beautiful landscape, a beautiful app, a mobile app that student can use and get necessary information that they need to deal with life's challenges. TOA Online is another such tool where you can get online therapy, but it is purely for students. It's not meant for grown-ups, so it's very much customized for the students. And at Ilumu, we are developing or designing a new product, a very advanced product. We call it a modern journaling, and you can use modern journaling to get help that you need. This is an explanation of what modern journaling is. We combine three different things. We call it self-concept clarity, artificial intelligence, and creative storytelling. These three are the main components on which modern journaling is developed. It is modern because it's in an app, and you don't have to text it. You can use your text, you can use your voice, you can use your video. Any way you want to journal, you can interact with these two characters. We call them Coco and Nora, and get the help that you need. Additionally, there is an artificial intelligence algorithm at the backdrop where it understands what your emotions are. As you interact with the app, it understands the words you are using, the preposition that you are using, how you are forming a sentence, how you are forming the grammar within a sentence, from which it can understand what your emotions are. If somebody is using voice, we understand their voice modulation and understand what their emotional state is. If somebody is doing video, we can look at their facial expressions, micro expressions on their face, and understand what their emotions are. Additionally, we can find out if somebody is experiencing depression, anxiety, suicidal thoughts. All of these are captured by AI to put together a much better profile of who you are, what you are dealing with, so that you can go and resolve these issues either on your own or with us or with the people you feel comfortable with. So that's what we are doing at Ilumo. How do we serve? We generally partner with schools, colleges, and universities to deploy our product. We don't really send it's not an app that a student can download, because there is partnership that happens within schools and colleges. We put together a profile for the school or college, and you can work with your school counselor or college psychologist so that our product has the maximum impact that you would need. So that's my brief presentation. I would be happy to answer any question that you may have.
Thank you so much, Saurabh Sengupta, for your wonderful presentation. Now it's the time for question answer session. The question for you, the first question for you from Dhritiman Chandaveri. His question is, how we the teachers can help out the students going through loneliness and indecision, which are nowadays regarded as a main reason of stressed students?
So, this is not necessarily something that teachers can resolve on their own. This is something where students, teachers, and parents, it's a three-party equation. So generally how we deal with it in our app is we created these characters, Coco and Nora, through which you interact with a lifelike involvement. It feels like a three-way chat, group chat with your friends. That reduces loneliness quite a bit. You don't feel that you're alone in this journey. And on top of it, we put together this dashboard for the teachers or school psychologists to look at, where they can look at it on a preventive basis and see that somebody is scoring high on loneliness, so they can partner with the student and ask what's happening in their life. Why are they feeling lonely, as it is indicating in the dashboard? And then if need be, the teacher interacts with the parents to get a better context of what is happening around loneliness.
Your next question is coming from Professor Debolina Shaha. It's very interesting. Sometimes we see that many of our students can't share their view with their parents. It may be because of their generation gap. They feel lonely then. They sometimes want to share with us, but sometimes we also can't help them individually. Gradually, they lost their interest in studies, and sometimes they shift into depression. Her question is, in that case, as a teacher, how can we motivate them to come back to normal life? And she has also another question. Do you think that academic institutes need to arrange mental stress reducing workshop for students?
I'll answer the second question first, because that leads to the answer of the first question. Yes, I think academic institutions need to take mental health seriously. As I mentioned, in my presentation, learning does not happen in isolation of the overall well-being of your brain. If your brain is not in position to learn, it doesn't matter how beautifully we are teaching. It's not reaching anywhere, right? So mental health is an important part of the academic setup as well. So that's the first part. Now, whether how would you enhance mental health is a completely different question. You can enhance it through webinars. You can enhance it through apps, as I mentioned. You can enhance it through some courses, lectures that you can conduct in your schools. There are many ways you can enhance mental health, but you cannot think of mental health as an isolated problem from academics, especially for students. If they are not doing well mentally, they are not in position to learn, no matter how beautifully you are teaching them. The first question is, you are absolutely right. That's the traditional arc that we see, where somebody is stressed and the situation is not really that worse, but they are getting isolated because they are feeling generation gap with their parents. Then they start interacting with the teacher, but the teacher cannot offer solution to all the students. So student-teacher ratio becomes a big problem in such circumstances. And that's one of the reasons why I think we need to incorporate AI in this entire equation. No matter who a student is, no matter how unique their challenges are, you would always see that there is a cluster of students who are feeling similar. If you can break down a group of 1,000 students into, let's say, 20 clusters, you are essentially dealing with 20 different programs. You kind of develop these 20 programs, and you take care of all 1,000 students, but for that you need data, you need to understand who is going through what. That's why a digital platform is a lot easier to deploy customized program for the students. So I would definitely think that AI-powered customization is the right solution, unless you can increase student-teacher ratio, which I think for many cases it's impractical. There are only few private schools in US that can afford such high student-teacher ratio. Otherwise, digitalization is the tool that we should leverage.
The next question for you is coming from Professor Abhishek Dash. The question is, can your system partially replace the need of therapists?
Partially, yes. As I said, we have about 50% of the student population that are going through severe stressors in life. We cannot replace completely, because it's madness to think a machine will replace human anytime soon. Not happening in the next 100 years. But in many cases, student situations are not such that they need to go see a therapist. If you can actually do an early prevention, you can resolve the problem digitally. So I think about 30 to 35% of the problems you can resolve digitally without really seeing a therapist. Another 15% of the issues that students are experiencing, for them you need a therapist. You need a human to intervene and offer the solution that a student needs. So I would say among the 50% students that are experiencing severe stress, about 30 to 35% can be resolved digitally. You don't need human intervention for them.
The next question is coming from Antariksh Venkataraman. The question is, can your system or a psychologist be a better alternative than antidepressant? As we know, they can be harmful.
Right. We don't know that yet. That is a question for which we need to do a clinical trial. At least our program is currently in an AI learning phase. AI goes through many different phases of production. It is a learning phase first, and then you deploy it, then you keep learning it, right? Our is in a learning phase right now. Once the learning is over, once we deploy it, then we are going to do a clinical trial, and that clinical trial data can give us concrete understanding of how do we compare with antidepressant. But we need to understand something about antidepressant itself that goes to my initial bifurcation on what is depression, right? Depression can be something that is inherent, that is a mental illness, or depression can also be something that is momentary due to some stress that you are experiencing. In my view, psychiatrists don't necessarily start with antidepressant if the depression that you're experiencing is a momentary, if the diagnosis is not clinical depression, generally they don't offer antidepressant. So our problem generally focuses on the people that do not have mental illness depression. They don't have clinical depression. They have generally the life stressor that that has resulted into a depression. That we resolve. We don't resolve the clinical depression. So we are not truly comparing against antidepressants.
Thank you so much, Mr. Sengupta. For time constraints, we can't keep this session for long. We will share the email ID of Mr. Sengupta. If you have any further question, you can get connect with him. Thank you so much.
Great. It was an honor. Thank you.
It's the time for our next session. We are honored with the presence of our next speaker, Dr. Indrajit David. Dr. Dev has completed his PhD from Department of Biophysics and Molecular Biology and Bioinformatics under University of Calcutta. He had been a visiting researcher in Department of Biosciences and Nutrition in Karolinska Institute. He has also done an international research fellowship funded by the International Center of Genetic Engineering and Biotechnology from Trieste, Italy. Dr. Dev is now associated with the Department of Biophysics, University of Michigan. He has many publications published in several renowned journals. The topic of his presentation today is accelerating rare dissociative process in biomolecules using selectively scaled Md simulation. Over to Dr. Dev.
Hi everybody. I think am I audible?
Yes.
Okay, great. Thank you, Chaitanya, for the nice introduction. And so I'm going to share my screen for today's presentation. Can any one of you please confirm that you could see my presentation?
Yes, we can see.
Okay, great. Well, good evening, everybody. Hope you guys are all doing well during this time. And as Dr. Sengupta mentioned, like we are really in a very stressful time when there is a coronavirus pandemic, and we are in stress in several ways. So during this pandemic time, bringing up this kind of webinar, I would say definitely help. This is helpful for all of us. Anyways, so it's a nice sunny morning here. It's a bit chilly, a little bit in the Midwest of America. It's 17 degrees centigrade. Well, so let's start our presentation. So this is a work of developing an enhanced sampling simulation approach based on Md simulations. And the Md simulations are this kind of method is very popular in computational chemistry and computational biophysics to study biomolecules. But the problem is whether that Md simulation data can be made useful to accelerate rare dissociative processes in biomolecules. And the Md simulations are in general known to be a tool to study the dynamics in a system. And generally, people like early researchers, they do visualize the simulated trajectory in a visualization window. But the question is how we can make use of the high-dimensional Md simulation data to explain biological phenomena. So that is the major question I would like to answer in my today's talk. And I will also, like to tell you that this work has been published last year in 2019 in SES Journal, Journal of JCTC Journal, Journal of Chemical Theory and Computation. So if any one of you are interested to go into the details of this method, please scan this QR code that will direct you to the article. Well, So the motivation of this work was to study biomolecular dissociative events that play a critical role in many cellular processes. Now the question is, when I talk about biomolecular dissociative events, then let's have some example of biomolecular dissociative events. So for example, the nuclear transport of precursor microRNA. So precursor microRNA is like it gets synthesized in the nuclear compartment, and after getting synthesized at the nuclear compartment, that precursor microRNA needs to be transported to the cytoplasm so that it can further be processed to become a mature microRNA. Now the question is, like, I mean, people from the experiments, they already know how it happens, like how the transport happens. Like the precursor microRNA, after getting synthesized, it forms a ternary complex with its associated protein RAN, which is bound to the GTP, and the nuclear transport protein exportin 5. And once this ternary complex gets synthesized, then it passes through the nuclear pore complex that is attached to the nuclear envelope. And once it transported to the cytoplasm, then a single GTP hydrolysis takes place. And that single GTP hydrolysis trigger the dissociation of this ternary complex so that the free precursor microRNA is delivered into the cytoplasm. Now it will be get processed to become a mature microRNA. So here the question arises, how a single hydrolysis, which is far from the binding site of the precursor microRNA and exportin 5, how this single hydrolysis could trigger a network of interaction so that this dissociation of the ternary complex could happen. And also here the question is, like, what is the time scale of this kind of dissociative events? When I mean, in my title, I called it rare dissociative events. We call it rare because this kind of dissociative events happen in the order of millisecond or second time scale. Now the question is, how using theoretical tool like Md simulations, one could capture these dissociative events, because Md simulations are computationally super expensive, and simulating this kind of cellular processes using Md simulation can be very computationally expensive. So our method is a method of enhanced sampling, which tried to capture this type of dissociative events. And also after capturing that, how we could use that data to explain biological phenomena and atomistic details and mechanistic details of these kind of processes. How we can do that, I will try to explore all those aspects in my talk. Another important dissociative events is associated with the drug discovery. For example, in case of CDK2, like cyclin-dependent kinase 2, which is already known as an anti-cancer drug target. So interestingly, there is a small molecule called ronicislev. This small molecule is already in a phase 2 clinical trial as a anti-tumor small molecule, and it has been found that the IC50 from the experimental data, experimental study, and the free energy of binding of this small molecule has already been explored in vitro. Interestingly, another analog of this small molecule have similar IC50 value and similar delta G value. Now the question is how one could differentiate which is more potent, which is more efficient as a drug molecule. So, and this question arises because this IC50 or delta G, these values are in vitro data, in vitro parameter. And in an in vitro system, these kind of parameters are not reliable when we compare the efficacy of these drug molecules in vivo. And this is because in vitro system, there is always equilibrium. But in real life, in real system in vivo, there is not such an equilibrium. There is always a flux going on. So how one could model the efficacy of this kind of system, and how one could study like the unbinding or the binding mechanism of this small molecule. In this regard, there is a parameter that is called residence time. So residence time is like a parameter which measures the drug efficacy. And it has been found that the residence time can be determined from surface plasmon resonance spectroscopy experiment, and residence time is a parameter which could reflect the in vivo drug efficacy. And here you see the residence time of rodinsilicate is like 10 times higher compared to its analog. And here you can see like the residence time data clearly shows how the rodinsilicate showing the superior anti-tumor efficacy in vivo. So residence time is simply a inverse of the off rate of the binding unbinding chemistry. And it just like if you know the experimental off rate, or if you know somehow can predict the unbinding time, then you can easily determine its residence time. So let's see if we could make use of Md Stimulation study to predict the residence time to rank between different small molecule targeting protein, RNA, or also if we could make use of the Md Stimulation to study other unbinding events. So the question is now, what is the molecular dynamic simulation? So the molecular dynamic simulation is like, as you know, as I already told you, the classical molecular dynamic simulations are very well suited to study the dissociative processes. Like, what is it actually? So actually, Md Stimulation is a computational microscope. In some words, we can say like that. It's a computational microscope. Like experimental biologists, they use microscope to study chemical, cellular processes. Like they inspect how the processes are happening in cell. So Md Stimulation is such a microscope, but here, rather than using a physical microscope, we use computational tools. So if ideally one could simulate a system in a biological time scale, then ideally it could display the cellular process. Now, that is the big question. If we could simulate a biological assembly or a system in a biologically relevant time scale in the millisecond or in the second order. So here, a very interesting information I want to provide here, the Anton supercomputers that are generally, that is developed by the D. Shaw Research Group in New York. That Anton supercomputer was named by the self-taught Dutch scientist Antonia Philips van Leeuwenhoek in 1600th century, who is known to be the father of microbiology. And who is like the early inventor of the microscope. And these Anton computers are developed by the Isha Group, and these are currently known as special hardware which could simulate biological system in millisecond or second time scale. Next, what is the mathematical background behind the Md Stimulation? So Md Stimulation is, by definition, it simulates physical movements of atoms and molecules in a biological system or any other chemical system. So what it does, it actually solves Newton's equations of motion. So according to the Newton's second law, so a force acting on a system or acting on a particle is the multiplication of its mass and its acceleration. So as we know, like force is equal to mass into acceleration. And on the other hand, force can be also determined from the negative gradient of the potential energy of the system. So if we combine this first two equation of force, then we could get this third equation, which is a differential equation. And if we numerically solve this differential equation, then we could get the position, the new position at different time intervals. For example, here, like initially, we need to define the initial positions of the atoms, like the coordinates, the crystal structures or NMR structures, or any modeled coordinate, and we need to assign the velocity of the atoms, and the velocity assignments can be done from simple Boltzmann distribution of velocity. And then once the initial positions and initial velocities are known, then we calculate the forces by from the derivation of the potential energy. So once we know the force, initial position, and velocity, then we solve numerically the Newton's equations of motion at different small time intervals to get the new position and new velocity. So, and then we repeat these steps iteratively to get a trajectory of motions of the system. And here I should mention that these small time scale like the delta t should be in the femtosecond order to capture the motion involved. I mean that is actually relevant because the femtosecond time scale is the fastest motion in a molecular system, like the bond vibration. So here you could see like when the biological time scale is like millisecond or second, then we start integrating numerically at the femtosecond order. And like you see how many thousands and millions of steps we need to do to calculate, I mean, the new positions of the atoms. And this actually is very computationally expensive. Even with current power of like parallel computing and graphical processing unit, when we simulate like thousands of like 100 residue RNA or protein system in explicit solvent, then it becomes like 50,000 atoms. And for each atom, you need to calculate the energy and you need to solve the Newton's equation. So that actually takes that much of computational wall time. So now the question is how we can calculate the. So here in the previous slide, I was showing the force can be calculated from the derivation of the potential energy. So what is that potential energy? So the potential energy can be calculated in many ways in theoretical chemistry, like using quantum chemistry or classical mechanics. So using molecular, and as we are talking about classical molecular dynamics, so here the potential energy will be calculated by a mathematical function, potential energy function that we called force field. So in that force field, actually there are a couple of energy terms that are actually additive, like the bond potential, then angular potential, and then the torsion angle. And there are two terms that is associated with the non-bonded interaction. So the first non-bonded interaction is the van der Waal interaction. That is actually in force field, that is actually approximated by 612 Lennard-Jones potential. And another non-bonded interaction is electrostatic potential, that is approximated by a Coulomb interaction of the partial charges of the atoms. So this is the simplest form of a force field mathematical equation. There can be additional terms like improper and Urey-Bradley terms also that can be associated, but this is the basic simplest form of a force field to calculate the energy of a molecular system in classical way. And later you will see I will mainly focus on this non-bonded term where this Lennard-Jones potential, we will play with this Lennard-Jones potential selectively in our enhanced sampling simulation approach. I will come into that later. So now let's see how difficult it is. Like what is the challenge between the mismatch between MD simulation and biological timescale. So we did MD simulation of CDK2 bound to Ronesis-Sleeve small molecule, and we did the simulation for like 20. We did the simulation for 20 independent simulation we have carried out. Each simulation was 250 nanosecond long, and each simulation was 250 nanosecond long, and each simulation took 115 hours. So, but we never see the unbinding of the small molecule from this from its active site. When we extrapolated this simulation time to its experimental residence time. So experimental residence times indicate the experimental, the time for that long, the ligand is bound in its binding pocket. And the experimental residence time is like 7,476.6 seconds. If we want to simulate this time scale, it would take 3 into 10 to the power 8 years, so which is a astronomical number. And in our lifetime, with our current computational power, it would never happen. Like we will never be able to capture this unbinding event. So this is the challenge. And let's see what are the enhanced sampling techniques currently available to overcome these challenges. There are many techniques that are available. For example, conformational flooding, metadynamics, adaptive biasing force method, steered MD, accelerated MD, selective AMD, then tau random accelerated molecular dynamics, and also a very latest approach that is called scaled MD. So scaled MD is the approach which we targeted because it is the latest and it is the simplest approach to implement, because all other methods need some expertise, and also it's very tricky to choose parameters for the other enhanced sampling techniques. So what is that scaled MD simulations? The scaled MD simulation is a very simple enhanced sampling tool which actually relies on scaling down the whole potential or the whole energy of the system by multiplying some factor with the total energy of the force field equation that I showed before. So here you see, like if it's a two-well potential, like one is bound, another is unbound, and by scaling the whole potential, actually we could lower the energy barrier between the two states. And eventually, by lowering the energy barrier, one could travel from one state to another state very easily within a couple of nanoseconds. Otherwise, if the energy barrier is high, then the conformations will be trapped in the local minima, and we will never see it crosses the energy barrier to the unbound state or other state. So this is the scaled MD approach, but also, again, when there is an approach, there are some limitations and problems. So let's see what are the issues associated with the scaled MD approach. There's a scaled MD approach. When we apply it, then actually what happens, the ligand unbinds, but the protein unfolds. And this is because, this is very natural, because we are actually scaling down the whole potential. And when we are scaling down the whole potential, actually we are scaling down also the non-bonded interaction potential between different residue of the protein. And as a result, eventually the ligand will unbound, but also the protein will unfold, because there is no force acting or weak forces acting to hold the secondary structure and tertiary structure of the protein. So what we need restraint. So during the simulation, what we could do, we could apply positional restraint or conformational restraint to the rest of the part of the protein, and we could apply some flexibility inside the binding pocket residues so that the protein doesn't unfold, but the ligand comes out. But the limitation here is when you are applying constraint or like different types of constraint, like conformational restraint or like positional restraint, actually you are restricting the natural motion of the protein. So these were the limitations in the previous scaled MD approach. We tried to overcome these issues in our approach. So what is our objective based on that? Based on that, our objective is to perform unbinding simulation without any restraint. So we will not allow any restraint so that we will restrict the physical motions or physical interactions inside the biomolecule. And what we could do after that, if we will try to see like whether we could recover the thermodynamic and kinetic parameters from the simulated data, and we could make use of it. And also, we want to study the microscopic details, like the atomistic details of interactions. And the strategy behind is like, rather than scaling the whole potential, we want to selectively scale the specific potential energy term in the force field. As I was showing in the previous slide, like the total potential, and there is a non-bonded term which is associated to the van der Waals interaction scale, approximated by 612 Lennard-Jones potential. We will specifically target that non-bonded interaction. And how we will do that? We will just selectively scale the ligand and the water molecules, like the interaction between the ligand and water molecule only, keeping the all other interaction as its place. So what we expect? We expect that if we scale up the interaction between the ligand and water only, that actually it means more and more water will try to interact with the ligand, and essentially the ligand will flood the binding pocket and wash out the ligand from the binding pocket, and keeping the physical motions and dynamics of the protein at its place. Let's see if we could achieve it. So here is the typical 612 Lennard-Jones potential. So here, if we multiply a simple factor with that Lennard-Jones potential term in the force field equation, actually we are scaling this epsilon, and this epsilon is the actually well depth of the Lennard-Jones potential. So if we use different scaling factor, we could lower and lower more and more the epsilon, like the well depth, which means that like at a particular distance here, more and more water will be favorably interacting with each other. So that actually theoretically it's valid. Let's see practically if we could do that. So when we tried that approach, like allowing more and more attraction between ligand and water only, we saw this simulation. Like you see here, initially the ligand was bound and the water went inside and it's surrounding the ligand molecule more and more, and the ligand is coming out, and we didn't restrain the protein and any other interaction. And we found that within 1.5 nanosecond only, we were able to see the unbinding. So here we have our initial success. Like our selectively scaled MD approach were able to accelerate the unbinding. Now the question is, when we have that success, can we use that scaled MD data to recover the kinetics and thermodynamics at the unscaled potential? Let's see how we can do that. So that's the objective. So how we could do that? For doing that, we actually take the help of Cramer's based rate extrapolation approach, where we use the Cramer's rate equation. Cramer's rate equation is a well-known equation and well-known approach in chemical kinetics, where this equation is applicable for like a two-state model, like the fast passage time. It's a fast order transition from bound and unbound state, or between two states. So what we could do from this equation, you can see these alpha values. These alpha values are the scaling factor that we are using. And then this tau alpha is the unbinding time, like how fast it unbinds from the binding pocket. So from our simulation, we have this tau alpha, we have this alpha. Then we simply fitted or extrapolated this equation to calculate the tau 1, which is the residence time or the unbinding time when we don't scale the potential. So we are predicting this tau 1 just by fitting this equation using our simulation data, alpha and tau alpha. And here the delta H, which is the enthalpy of the system, that was randomly chosen from a large range of values within the range of experimental delta H value. So let's see how it goes. So here you see, like, this is these black dots. These black dots are the unbinding time at different scaling factors. So you see here, like if we increase the scaling, I mean the unbinding time is increasing. Actually, we could say in other way, like if we scale the potential more, the ligand will unbind faster. So here you see here, I mean, from different scaling factor, we could see the relationship between the scaling factor and the unbinding time. This is an exponential equation. I mean, exponential relationship. So what we did, we fitted these 3-4 points and extrapolated to the scaling factor of 1. When the scaling factor is 1, that actually it's an unscaled potential. So simply by fitting the unbinding time exponentially, it's a weighted fitting, and we could recover the unbinding time at the unscaled potential. Now you see, let's see if we compare couple of small molecule, can we reproduce the correct trend of unbinding time that is experimentally observed? So we took these 3 small molecule, and we found that these 3 small molecule have the residence times of 6 second, 763 second, and like 7,476 second. So, and from our simulation, scaled MD simulation and extrapolation, we found that the similar trend, like 40 nanosecond, 10 to the power in the order of 10 to the power 4. And 796 and 8,588. So we found that from our scaled MD simulation, by extrapolation, we could recover the correct trend of the kinetics. Here is a big difference of the time scale, because the experimental time scale was in second, but our time scale was in millisecond. So we are 3 order behind, and this is happened because although we could recover the correct trend, the time scale didn't match correctly, and this is because we applied the Cramer's equation, which is applicable to the first passage time, like the initial unbinding, not the total, not like the full unbinding when the ligand unbinds and goes to the bulk solvent. We didn't consider the full unbinding. We considered only the first passage time, the initial unbinding. Now let's see if we could recover the thermodynamics. So to recover the thermodynamics, we have a free energy extrapolation approach, where actually one could calculate the free energy at the scaled potential, and the alpha values, like the scaling factors, are known. So this is a simple linear equation. So here the alpha values are known, like the scaling factors are known, and for each scaling factor along the unbinding trajectory, one could calculate the free energies just from the fraction of native contacts between the ligand and the protein, and taking that fraction of native contacts and feeding it to the Boltzmann equation, one could easily calculate the free energy. So, and then from this linear extrapolation, one could easily recover the free energy at unscaled potential. Let's see how it works. So here, like the 4 color lines or the plots, we will say these are the unbinding trajectory, or the free energy profile for the 4 scaling factors we used. And then at each point, these are the free energies at each fraction of native contacts. So, and you see, it is simple, a linear extrapolation. So at each fraction of native contacts, we fitted these 4 data points to recover the free energy at the unscaled potential. And when we did that, along the whole trajectory, we have a whole free energy profile at the unscaled potential, which is the black line. And when we have that unscaled free energy profile, then from the maxima and minima of this profile, one could easily recover the free energy of unbinding. And let's see if we could recover the free energy of unbinding and experimental free energy of unbinding. And here you see the same 3 molecules for which we were able to recover the kinetics. You see here the free energy of unbinding. Again here, we were able to recover the correct trend of experimental free energy of unbinding. And here it is much more accurate. Only there is a 2 kilocalorie of difference between the predicted and the experimental free energy of unbinding. So here, this way, using the protein ligand system, we were able to capture the unbinding. Also, we were able to make use of the scaled data to recover the kinetics and thermodynamics at the unscaled potential. Now, what are the potential advantages of this method? So the potential advantages are, it is very effective to accelerate the biomolecular dissociation. And then it could recover the kinetics and thermodynamics at the unscaled potential. It does not require any conformational restraint. It is a very general approach. One could easily scale up and also scale down. On the other hand, it does not incur much computational cost because the unbinding happens within couple of nanoseconds, and then it doesn't require any special scripting or change in the source code of the simulation packages. It can be easily implemented in CHARM, MD, GROMAX, and OpenMM, all the renowned open source and licensed version of the MD simulation packages. What are the limitations? The limitations are still a little bit of computationally expensive. You cannot screen like millions of small molecules during the drug discovery. With our current computational resource, one could screen like hundreds of small molecules maybe. So then we would suggest to use this kind of approach at the latter stage of the drug development. So these are the limitations. And also, there is also another limitation, that is always there is a big question when you are using a force field, then what is the accuracy of the underlying force field? So this question always remains there, and a bunch of scientists in our field, me also, worked on force field parameter development and optimization and validation. So, and then the approaches, like the applications of this method to the other system. For example, we tried RNA ligand complex. So the proof of concept for our method development that is published, that was for protein ligand system. Then when we got interested to apply this approach to the RNA ligand system, and also like other assemblies like protein nucleic acid complexes. So let's see what happens in case of RNA ligand complex. So we tried RNA ligand complex, and for that we chose FMN riboswitches, because FMN riboswitches nowadays is a structured RNA element which is druggable, and like the drug discovery scientists, they are targeting FMN riboswitches as a RNA drug target, and that FMN riboswitches, we all know the biological implications of FMN riboswitch, like how it works. Like when the small molecule binds to the aptamer domain of the FMN riboswitch, actually the expression platform, there is a change in the conformation, and the and either the gene will be expressed or will not be. And this kind of FMN riboswitches are very prevalent in bacterial system. So that's why these FMN riboswitches are nowadays a current target for for treating bacterial diseases or other implications. So when we applied our SSMD approach to the FMN riboswitches, we saw that we could also accelerate the unbinding of the small molecule from the FMN riboswitch FMN complex. And we were able to see the unbinding within only 30 nanoseconds, whereas this unbinding of, this kind of unbinding event could occur in hours or second time scale. We were able to recover the unbinding in 30 nanoseconds. Let's see how we can make use of this approach for RNA ligand system. So we chose two small molecules. One is WG3, the top one. Another is ribosyl B. So these two small molecules we chose because ribosyl B is the fast known FMN riboswitch targeting small molecule, synthetic small molecule. And WG3 is another newly synthesized small molecule that target FMN riboswitch. The interesting fact is that both of the small molecule selectively target FMN riboswitch. But even if both of are very selective, WG3 is biologically inactive. It cannot inhibit the bacterial growth, whereas the ribosyl B it can inhibit the bacterial growth. When we applied our SSMD approach to these two complexes, we found two distinct unbinding pathways. And we found that in case of WG3, like here, when the unbinding is in the unbinding pathway, like the exit 1, there is a high water density. Whereas in case of ribosyl B, the water density was around the exit 2 pathway. And we have identified two specific residues. One specific residues like adenine 48 here, which acts as a gate. Like this residue acts as a gate in two types of small molecule. So this also gives us a mechanistic detail like how in FMN riboswitches RNA mutation studies can be helpful in case of drug discovery. Again, but these are mechanistic detail. Let's see if we could make use of our scaled MD data to explain whether why WG3 is biologically inactive or whereas ribosyl B is biologically active. So for that, what we did a kinetic modeling. So from the scaled MD simulation, you see here we found like the unbinding time for WG3 is much lower. Like the WG3 unbinds from the binding pocket much more rapidly compared to the ribosyl B. And when we did the kinetic model, then we found the K-off values. The K-off values, if you carefully notice here, you see that in case of WG3, it is higher a order more than in case of ribosyl B. So it is 10 to the power in the order of 10 to the power 3, and here it is in the order of 10 to the power 2. So which indicates that even if they bind selectively, like K on are very similar, the off rates are not similar. The off rates like is higher in case of WG3, which indicates that in case of WG3, it unbinds faster. And when it unbinds faster, then it cannot lock the aptamer conformation so that the transcription termination could happen in the expression platform. And when we did a simulation of the ligand unbinding, we found that in case of WG3, the fraction of native contacts became almost close to 0 within 70 seconds. Whether in case of ribosyl B, the fraction of native contacts at 70 seconds is still around 60%. So we took this sensing window of 70 seconds because it has been found that experimentally, like the transcription of the aptamer domain of the FMN riboswitch, the time difference between the transcription of the aptamer domain of the FMN riboswitch and the expression platform, the time difference is around 60 to 70 seconds. And we found that within that 60 to 70 seconds, WG3 is initially it was bound and then it became unbound so that it cannot freeze the conformation of the transcription of the expression platform, whether ribosyl B was still bound there to do it work. And that explained that with the aid of our data, that why WG3 was biologically inactive and ribosyl B was biologically active. And now another application, like if we could use our scaled MD approach to study the disassembly of nuclear transport complex that I showed at the very beginning. So what we did, we applied here a little bit different approach. Like here, we didn't multiply the scaling factor with the whole Lennard-Jones potential. We didn't scale down the epsilon. We actually multiplied the scaling factor with the R mean. So the R mean, when we multiplied some factor, a positive factor with R mean, actually the minimum distance between two atoms in a Lennard-Jones potential that goes far and far. So that means the atoms will get stabilized in a further distance. Initially, it was here at the unscaled potential. So by this approach, actually, we are applying a repulsion between the atoms. Actually, then the components here, the exportin 5, RAN GTP, and microRNA, if we scale up the R mean between each pair of these components, then actually each component will see each other as a rival and they want to go far from each other and eventually they would dissociate. So that was our logic. And when we applied that in real, we saw that great, like these three components are actually dissociating and the microRNA is getting out from the ternary complex into the bulk solvent. So still, this is a proof of concept. We saw the initial dissociation. Now we are trying to make use of our data, high dimensional MD simulation data, so that we could explain the mechanistic detail, the network of interaction, how this unbinding triggers due to the GTP hydrolysis. The future direction of this work would be the studying the association dynamics. For example, if the ligand is in the bulk solvent, we don't know the binding pocket. So then we could apply repulsion between the bulk solvent and the small molecule. Then the ligand will not want to stay in the bulk solvent. It will try to find out a hydrophobic core. And as we know, inside RNA molecule, the binding pockets are hydrophobic. So ideally, what it should do, it should find out the hydrophobic core inside the binding pocket, and it will eventually go there and sit there, and we could study this association dynamics. This kind of studies has already been performed by DSO research group, but they didn't apply any enhanced sampling approach. So we want to apply our enhanced sampling approach to study this association, so that we could see the unbinding dynamics within a possible within couple of nanoseconds. So this is our future direction. We want to study this, and yeah, that's it. And yeah, thank you. I want to thank my research group here at the Frank Lab at the University of Michigan, Professor Aaron Frank, my PI, and my lab mates. Also, I want to thank the organizers of Spectrum of Advanced Biological Sciences, IQAC, and Department of Microbiology, St. Paul's Cathedral Mission College, Kolkata, India. And one interesting thing is that when I was planning to prepare for this talk, then I was remembering my old days when I was doing my bachelor's in Bongovashi College, actually in chemistry department. Then I went to St. Paul's College several times with my friends, and in the student dormitory, and I spent many hours there. With them. So it was. I was remembering those old days. Yeah, thank you, the organizer, for inviting me. It's my pleasure to give the presentation. And now I'm open for any question. Thank you.
Thank you so much, Dr. Dev, for the wonderful speech. You have a question from Professor Kumar Singh Roy. Generally, water is associated with the maintenance of the structure of different biomolecules. Does the presence of water molecule anyhow affect MD simulation? If yes, then how?
Yeah, that is a very relevant question, and that is the major challenge nowadays, because in case of protein system, or even in case of RNA system, like the protein or RNA residues are charged. Mainly in case of RNA, the backbones are highly negatively charged. Whether in case of protein, some residues are charged, some are not. But these water molecules actually sometimes, in case of RNA, it forms hydrogen bond with the backbone that stabilizes the particular tertiary structure or the secondary structure. In protein also, and sometimes in case of ion, sometimes there are ions associated for the stabilization of secondary and tertiary structures, and those ions could make a bridge between water, water ion residue. These kind of bridges are very important. So for the sake of electrostatic interaction associated to ion, these water molecules, like hydrogen bond, like water bridges, these water molecules are very important, and that's the reason people are nowadays more and more doing explicit solvent MD simulation rather than doing implicit solvent. Because in implicit solvent, you don't explicitly allow water molecules. Actually, you just allow some dielectric continuum dielectric constant that should replicate the water environment, but it will not replicate the physical atomic interaction, non-bonded interaction between the water and the biomolecules. So water molecules are, for that reason, water molecules are very important for MD simulations of biomolecules. In case of lipid system like PLP layer, that's a bit different, because also in that case, the water molecules should be properly modeled so that it could differentiate between the hydrophobic core of the PLP layer and the hydrophilic surface. So yeah, I think that's the reason water molecules are very important in MD simulations.
Your next question is from Dr. Rana Odhikari. The question is, can we use recombinant fragments for the target proteins like active sites of enzyme, binding sites of receptor in linear peptide form instead of using whole protein for scaled MD studies?
If I understand properly, if I understood exactly what you wanted to ask, this is because, like, always it is good to use the whole protein, because you are actually incorporating long-range interactions and long-range secondary and tertiary interactions as well. So when you are using only the active site, not the whole protein, that actually you are losing many interactions, and there are a lot of studies where protein network of interaction, like allostery effect, long-range interaction in protein folding, and protein ligand interactions are associated. So ideally, it will be best to use the whole system rather than a truncated system. But due to the computational power, lack of computational power in many places, sometimes that is not possible. So one could use the truncated system or smaller part of the system, but the modeling, like how you are modeling that system, is very important. What kind of approximations, what kind of restraints you are using. So that, the modeler needs to be very careful on that.
Your next question is from Professor Abhishek Das. Can we also use to see the metal binding through MD simulation?
Which binding? Metal binding?
Metal binding.
Sorry, I couldn't hear you.
Can we use the MD simulation to observe the metal binding also?
Yeah, so you said metal binding?
Metal, yes.
Metal, right? METL.
Yes.
Yeah, sorry. Yeah, definitely. Because, okay, there is a challenge, because when you are talking about unbinding of metal, then the metal parameters, because in classical mechanics, the simulation largely relies on the force field parameters. So in general, for example, for monovalent cation like sodium, potassium, those parameters are well tested. Even if we go from monovalent to divalent cation like calcium 2+ or magnesium 2+, those parameters are still not well parameterized. So when you want to use MD simulation for metal unbinding, the modeler needs to be very careful about the parameters of that particular metal. Ideally, it doesn't matter it's a metal or it's any other chemical system or biomolecules. The scaled simulation approach is applicable to any kind of dissociative events. It could be any combination of components for any assembly.
You have another question from Shraddha Mahendra. Can this MD simulation will help us in disease prediction?
Disease, I'd say disease prediction, yeah, in some sense, because from the perspective of ligand, drug molecule interaction with the small molecule, if we could connect somehow this biological activity of that small molecule to the disease molecule, like where it binds, then it can be useful. on the other hand, directly disease prediction may not be directly correlated with the MD simulation, because disease prediction, I think it depends on different, it depends. Like if, for example, for the degenerative diseases like ALS or Fragile X, so in those kind of diseases, actually it's a RNA repeat sequence. So let's forget about ligand. Let's forget about drug discovery. Just consider a simple RNA molecule which has a repeat sequence, or which has like a uncommon or like unconventional base pairing or sequence pattern. So we could one could simulate if that system is stable or not, or whether this kind of sequences can be like this kind of sequences, if it is stable or not. So that stability information may give us some information whether that biomolecule is prone to be a disease molecule or not. So yeah, one could try to do that simply by simulating disease-prone small molecule, like unfolding. Like there are several diseases which are associated with the folding and unfolding of RNAs and proteins. In that case, in that sense, it should be useful. Yeah.
Thank you so much, Dr. Dev. For time constraint, we can't keep any more questions. But we all know MD simulation is very new and very challenging area for nowadays world. And we all have very little knowledge. We deal with undergraduate students. They also have very little knowledge about the MD simulation. But your presentation was really helpful. You presented in a very simplified way. So thank you so much.
Thank you. It's my pleasure. And all the students and listeners, everyone, you are welcome to email me, contact me if you have any question, if we could collaborate, if we could discuss. And so any one of you are very welcome for that. Thank you.
Thank you so much. We are going towards our next session. Our next speaker is Dr. Atiya Rahman. Dr. Rahman has done his BSc and MSc from Department of Biochemistry under University of Dhaka, Bangladesh. He has done his PhD in Applied Bioresources Science from the United Graduate School of Agricultural Science, Ehime University, Japan, and postdoctoral fellowship from the Department of Biochemistry, School of Biochemistry and Genetics and Microbiology under University of KwaZulu-Natal, South Africa. Dr. Rahman is currently working as a professor and chairman in the Department of Biochemistry and Molecular Biology in University of Chittagong, Bangladesh. He is going to present on the topic mechanistic outreach of functional foods to defend respiratory virus. Over to Dr. Rahman.
Hello, everyone. Can you hear me, please?
Yes, sir.
Okay, thank you so much. I'm so sorry that, in fact, I have another meeting, and I think I have a shorter time to spend here. So, Honorable Chair of the session, distinguished scientists, ladies and gentlemen, I'm very much delighted, and it's my great privilege to present in such a very enlightening session. I'm thanking the St. Paul's Cathedral Mission College, particularly the organizers, IQAC and Department of Microbiology, on a spectrum of advanced biological sciences. I'm so grateful to introduce me in such a way. So let me present. Actually, my presentation is not that much very high throughput science. Rather, this is very simple science, which we do feel that in such a pandemic and COVID situation, our knowledges and the outreaches should be in such a way to be designed that we people and the general people could be benefited. So what's my topic in Dutch cover? So I'm working on functional foods. And this presentation is one how the functional foods they do contribute in mitigating the problems and the infections and the invasions of the viruses currently we're talking about. So we are calling functional foods. So let me make a short brief on functional foods. If we could go back in 1980s, in that case, we'll find that Japan, they first introduced this functional food topic in mid-1980s. Before that, actually, science didn't explore in such a scientific form of functional food. Rather, we are very much accustomed to use different sorts of food, which would be very valuable and beneficial for different sorts of diseases and ailments to be cured. So, how they defined Japan and the Japanese scientists, they define functional foods as food which still have the benefits out of the gastronomic pleasures and they're very basic nutrient supply. And you see that before Japan, there is no legislation about the functional foods. And currently, functional foods is very much in vogue and very highly, you can say, the prospective ways to think how this sort of foods could be the beneficial sources. Therapeutic approaches even could be addressed using this sort of foods. And they have made the legislation of functional foods, they named as FOSH. And this is beyond Japan has been far reached to Europe, America, and some other parts in such a manner that they have the capability to improve immune function, they can prevent some particular diseases, and sometimes they can support to recover some specific diseases. Very prominently, scientists are focusing very interestingly against the aging procedures of a human being, how functional foods can help in this regard. So the historical background so far we have tried to focus. The next question is that what's the basic approach to explore the functional foods? As a scientist, or as a listener, or as a student, or the researcher, the very basic approach to explore functional food is the identification first. And that identification is the screening procedure. Through screening procedure, we identify new sources and we target some pharmacological or phytopharmacological effects. And based on that pharmacological or phytopharmacological potentiality, we standardize our food sources. For example, currently I am working on a Bangladeshi plant which has been also available in India, in some parts of India. We are trying to make it a very valuable source for anti-diabetic approaches. So that is turned into functional foods through some sorts of, you can say, the mechanistic approach through a hypothesis-driven human nutritional studies. What does it mean? It means that we first screen out, then we standardize them, then we check their nutritional values. I mean their minerals content, their vitamin contents, their some other minerals. After all that, if we find that even their in vitro study, their in vivo study, their in silico study, all together we find that this is non-toxic. In that case, we approach, yes, this food could be used in our daily manner in particular dosage and administrative ways. And finally, we prescribe that yes, that food could be very beneficial and that could be very much usable for particular sort of disease. So this is how we explore the foods into a functional food sources. Interestingly, currently, very small portion or a small percentages of the whole population, they do have idea about the functional foods. You see that almost 33% of the world population they have never heard about the functional foods. And only 20% people they have the idea about the functional food. It does mean that still there are ways how we can explore new sources of functional foods, and we can make the current existing sources usable for different sorts of diseases. And interestingly, currently the phenomena is such that in Europe and America, the trend is very much rising. I find that in USA, currently they are using this sort of functional foods in different forms, and that is the total market share is around $24.6 billion. Even in Malaysia, this is $3.7 billion. It proves that actually this is a highly prospective field where the scientific evidences could help get them usable and useful for different sorts of disease prevention and therapeutic approaches. So what are the basic ways and basic, you can say, categories of functional foods? The functional foods, there could be prebiotics, there could be probiotics, there could be vitamins, there could be minerals, there could be fatty acids, some other dietary fibers, and sometimes the phytochemicals, particularly the steroids, the carotenoids, the flavonoids, the alkaloids would have this sort of capacity, and they could be considered as the functional foods. So let me share that what are the major functional food sources. We usually highlight their function as antioxidant, then you can say anti-cancer, anti-arthritic, anti-proliferative, then hypocholesterolemic, then immunomodulatory, anti-hypertensive, neuroprotective, antimicrobial, anti-diabetic, all these sorts of diseases and pathophysiological conditions could be mitigated and addressed using the functional foods. So next question is what are the sources? In our subcontinent, our people usually use the plant sources. The plant source. However, if you go to Latin America and neighboring countries, you see they also use animal sources. Very interestingly, when I was you can say I was in primary school, I got some night blindness, and my maternal, I mean my grandfather, what he did, he offered me some banana, and he put some insects inside the banana, and he told me that take this banana continuously and successively 3 days, I think you will be fine. Very interesting that in my primary level, I got the night blindness. I didn't have any idea about the science. But once I came to study biochemistry, I came to know that that insect did have some sort of luminescence. And that luminescence triggers the visual cycle, where the 11-cis-trans retinal system could be triggered. And that is the way how that particular insect re-triggered and rejuvenated my night blindness. Very interesting. So this is functional foods. This is functional food. Sometimes people intermingle the functional foods, alternative medicines, then you can say herbal medicines, traditional medicines. Yes, many of the traditional medicines, they could be as well used as functional foods. However, all functional foods are not traditional medicines. All functional foods are not herbal medicines. All herbal medicines are not functional foods. So our functional food sources could be animal sources, there could be plant sources, there could be nutraceuticals, there could be some other symbiotics, some other industrial products, there could be functional foods. Hope you will be agreed with me. So next question is that what is the basic mechanistic approach we are going to deliver? Our basic mechanistic approach is that to trigger or boost the immune systems using the functional foods. As we know that our immunosuppression, immunocompromisation in different sorts of trauma or some sorts of infections, we lose our basic and standard immune procedures and immune capability, which is supposed to be combination of the innate and adaptive immune systems, where the innate immune system is always on. For example, our epithelial, our outer barrier of the skins, our mucosa or gut systems and all the others. Once they do fail, they seek supports from the adaptive immune systems, and they usually respond in delay. With the aid of lymphocytes, for example, the B lymphocytes, T lymphocytes, and different sorts of markers are involved in that cellular systems. All together, using the cell-mediated and humoral immune system, the immune system finally ensure that they are quite capable to defend the invaders. So functional food is our choice to make immune system ensure that they can defend the invaders in right way. That is our mechanisms, or that is our approach, what we are going to present indeed. So the very common term is that you boost our immune system. So what's the immune boosting? I have browsed the Google and I find that this is billions of the keywords regarding the boosting of immune systems. However, hardly will find how the functional foods they are related to boost up the immune systems. Therefore, we try to focus these functional foods efficiency and the potentiality how they can boost up these immune systems. The immune systems, immune boostings, it does mean holistically the immune balance. If the immune system can ensure the way it is supposed to be maintained in our immune system. For example, the pathogens, either it's from outside or from inside. Sometimes we call autoantigen. In all regards, if the immune system can ensure that integration of immune response could be the combination of your innate and adaptive immune system as we discussed. In that case, we'll find, yes, my immune system is quite balanced, quite immunoboosted. And to ensure that using the functional food is one of the targets. So you will be very much agreed with me that actually the immune function, it varies and depends on the ages, the physiological conditions, people natures, behaviors. For example, your food pattern and food behavior, your nutritional status. If you are malnourished, in that case, your immune system cannot support you indeed in the right way. Then if you are accustomed with smoking, it would be different from the person who doesn't smoke. If you do consume alcohol, it will be different. I mean, the immune function would be different from the person who doesn't take alcohol. If you are under stress, environmental stress, physiological stress, or psychological stress, if you are immunocompromised, if you do have some vaccination history, if you are exposed to some sorts of pathogens, if your hormonal status is somehow troublesome or defective, if you are younger, in that case, your immune systems the immunobalance will be different from the person who will be older. If you are male, your immune system's function will be different, might be, from that of the females. So holistically, the immune function is dependent on this sort of factors. And next question is that since we are going to use the functional foods, we have to know how the pathogens they do infect us. Since our target is the respiratory viruses, and currently since we are facing the COVID crisis, in that case, my talk will be confined on the viral infection particularly. So as you know that the infection of viral and the mortality, morbidity from the viral infection, it depends the nature of the viruses, types of viruses, and their type of infections. You know, if the viral infection or the nature of infection is abortive, that will be different from the viral infection that is productive. If the viral infection is productive, in that case, it will be different from that of the destructive. Some of the viruses, they do insert and they infect the cells. However, they integrate with the host genomes, but they don't make new progeny. But they could be survived long time. For example, HIV virus in the host cell. However, if we look into the COVID-19, I mean SARS-CoV-2, in that case, we feel the people are getting to be destructed. I mean the genomic system, people's whole DNA, RNA, and all other systems are going to be collapsed because of the infections. So the mode of viral infection is truly modulated by the viral infections. You see, many of the infections, they directly kill the host cells. Sometimes they do inhibit the DNA, sometimes they do RNA, sometimes they inhibit the protein synthesis, sometimes they integrate with the enzymes. They degrade the enzymes of the host cells and eventually they kill the host cells and people go to die. But in that context, the most important point is that why this sort of variations is happening. So far, we have the evidence that once a virus is infected, in that case, the balance of our immune system leads to cause the oxidative stress. What is oxidative stress? The oxidative stress does means on the person produces, or the physiological procedures or biological system produce the reactive oxygen species or reactive nitrogenous species. When they lose their oxygens and they turn into the free radicals. In that continent, the circumstances, we see the free radical production is getting higher than the capacity of the biological system to make the antioxidative potential in that particular system. If the system faces this sort of problem, in that case, we call that this is called the oxidative damage. And this damage, holistically, we call the destruct our genomic systems, our protein system, our nucleic systems, all together. That means we are very much becoming vulnerable to disease. Interestingly, the viruses which are currently in discussion, we see that most of this sort of viral infection, they cause to increase few of the enzymes. For example, number one, we call NOx, nucleotide nicotinamide adenine dinucleotide phosphate oxidase. Another enzyme is xanthine oxidase. Another one is dual oxidase. It's now evident that in most of the respiratory viral infections, the these three enzymes concentrations are very high. I mean, when the infections are caused, these three enzymes particularly increased in a very high level. And we know that once the free radical is getting increased, we are in oxidative stress, then multiple complications are happening. For example, our cardiovascular system, our retinal system, our limbic system, our nephrotic system, in a word, the holistic system are vulnerable in oxidative stress. Therefore, we see that once SARS-CoV is infecting somebody, in that case, people are having these three enzymes in high concentration, which lead to cause multiple organ failures sometimes. Today, we lost our ex-vice chancellor of my university. He was COVID positive, and his age is more than 65, around 70. After 7 to 8 days, very chronologically, we found that his kidney is not working, his lung is not working, like this. Then finally, he expired. The question is that how the functional food can help attenuate these three enzymes or the infections. So we see the functional food sources so far very much common in our country or some other countries. We are describing and suggesting the people during COVID infection to take ginger tea. We suggest people to use turmeric. We suggest people to take much citrus fruits. We suggest people to take almonds. Why is that? Because of the phytochemical properties and the composition as well as the phytoconstituents existed in this sort of fruits and functional foods. If we look into the properties of this sort of functional foods, phytoconstituents, most of them are the polyphenolic compounds. And these polyphenolic compounds, in our laboratory, we found that they have the strong potentiality and capacity to reduce the oxidative stress. We checked in vitro different sorts of model. We checked in vivo. Then we checked in silico model through some sort of simulation. Then we went to go for measuring the mRNA expression of this sort of enzymes. Interestingly, during COVID, we found that some chloroquine, which was once upon a time prescribed for COVID. And if you go, you see that the very common functional food is black seed, we call the Nigella sativa. If we analyze the phytochemistry or phytoconstituents of Nigella sativa, we find almost 3 or 4 derivatives of chloroquine. And sometimes people call this is Nigella sativa black seed. It could be the treatment for all diseases except death. Why is that? Because as I said, there is a strong relationship with the polyphenolic properties of the phytoconstituents which are working to reduce the oxidative stress. Those which can reduce the oxidative stress, they can reduce the enzymes increased in our respiratory viral infections. And that is proof. Let's see next. So you see the functional foods. This is many of the functional foods. We see wheats, citrus fruits, oats, rye, and many other fruits containing polyphenolic compounds. And we find they have a strong antibacterial activity, antifungal activity, antiviral activity. And through intense use of this polyphenolic compound, finally, what we find they are capable to reduce the oxidative stress. Why is that? The challenge is that to reduce these three enzymes. Interestingly, once we are able to reduce the oxidative stress, it does mean we are able to increase the concentration of superoxide dismutase catalase glutathione peroxidase. It does mean the relationship in between these three enzymes are vice versa. Once your biological system is vulnerable to virus, it does mean your superoxide dismutase catalase glutathione peroxidases, they are reduced. Whereas your NADPase oxidases, xanthine oxidases, then dual oxidases, they are increased. So we are trying to focus how functional food and whether functional food can reduce this three enzyme in the left side and can increase this three enzyme in the right side. You see, the curcumin. It's evident. I'm sorry that I forgot to put the references here, citation. So curcumin, very common in Indian subcontinent. We usually use with our vegetables in our daily cooking practices, curcumin. And we find that this curcumin, this is a very strong source to reduce the oxidative stress using that at least 4 subtypes of NOx enzymes to be reduced. NOx 1, 2, 3, 4. So once this curcumin is capable to produce a high capacity or highly the superoxide dismutase, then glutathione peroxidase and catalase, they will as well be able to reduce the NOx. And we find that this viral attacks once happen, in that case, as we said, that ROS, reactive oxidative species are getting highly triggered level. So this sort of triggering ultimately causes the death of the cell. So what they are mostly linked with the inflammatory pathways. They produce numbers of inflammatory mediators. They ultimately cause the cytokine production high. We call the cytokine storming. And that topic has been so much popular in this COVID situation. So our functional foods, the use of functional foods is indeed linked with reducing this cytokine production. And there are a lot of pathways. For example, as we said, the curcumin. We said that this curcumin, they are capable of modulating the NF-kappa B cells pathway that drives the transcription of other genes via activation of nucleoside and nucleoside binding. Particularly the leucine repeat, pyrin-containing domain, inflammasome in ROS-dependent manner. Again, we checked in our laboratory, as I said, that curcumin and with other Bangladeshi plant Leomacrophylla, that in particular organ, for example, liver, the level of catalase and superoxide dismutase, after administrating this sort of functional food sources, we find that multiple increase. It does mean they are capable of reducing the reactive oxygen. They are capable of increasing the catalase and superoxide dismutase. So that's what we were trying to find out. Because once catalase and superoxide dismutases are increased, in that case, NOx, DOX, and all these three enzymes are getting reduced. What more? The next phase is the pathways. Different sorts of signaling pathways are as well evident to be mitigated, or you can say the different sorts of pathways are linked and managed by the functional food sources. We see that many of the functional foods, they inhibit the viral integrase protein. They sometimes degrade the viral cat protein. They sometimes degrade some other viral genomic replication factors. They sometimes kill the viral some other proteases. So using this sort of functional foods, we again see, for example, the Nizala sativa of, you can say, black cumin. We found that in a cell line study, there are some proteins, for example, TRP1, TRP2, or the trypsin receptor proteins. At least there are 8 variants of these proteins, and this protein is very much important to produce the interleukin and cytokine, some other proteins. And we find that this TRPA1, TRPC4, TRPM6, TRPM7, these factors are increased very much, which are very much important to manage the respiratory tract viral infections. Some other pathways, for example, as we said, curcumin. This curcumin is found to prevent the viral attachment and entry by abrogating the function of the viral envelope. They as well modulate the host cell signaling pathways, for example, as we said, NF-kappa B cell, P13, AKT, and some other inflammatory pathways. Sometimes the curcumin serves as the biocidal agent by attacking the integrity of the viral envelope. So the question is that, are this all to kill a viral to kill a virus or to protect a viral infection? Indeed not. There are some other researches. They have shown that, for example, the coconut oil, this could be used as a viral the functional food. They found that the coconut oil, there are 3 mechanisms at least established, then they can cause the disintegration of the virus envelope. Second, they can inhibit the late maturation stage of the viral replication cycle. And finally, they can prevent the binding of the viral proteins to the host cell membrane. So these are the 3 mechanisms how a virus can get inserted in the host cell and make the environment to kill the host. So if we can manage, or if the functional food can manage these 3 mechanisms, so this would be enough in conjunction with mitigating the reactive oxygen species and reactive nitrogenous species, increasing the enzymes, as we said, and the enzymes decreasing, as we said. There are some other pathways. We see that there are some transcription factors. For example, the nuclear factor. The nuclear factor E2 related factor, we call them NRF2. So during the normal reactive oxygen species level, this NRF2 retains in cytoplasm with another protein that we call the KEAP1. So once this is in normal ROS level, they are integrated. But NRF2 dissociates from KEAP1 and translocates into the nucleus and binds the antioxidant response element of the target genes, encoding the predominantly antioxidant enzymes. So this is very interesting. Once this has happened, in that case, we found that NRF2 regulated target genes are superoxide dismutases, catalase, peroxidants, then glutathione peroxidants. These enzymes are triggered to be synthesized. If we can do that, in that case, the antioxidative capacity will be highly elevated. And if we can manage that, in that case, our reactive oxygen species would be reduced. So I would like to conclude my session, my talk. Holistically, the functional foods, they are highly capable using these sorts of modes we discussed. They can manage the inflammatory mediators, inflammatory factors. They can manage the antioxidative factors. They can manage the immunomodulators. They can manage the humoral and cell-mediated factors, which eventually ensure to protect the invasion of virus, particularly the respiratory virus. So many of the functional foods are currently used, and sometimes we know them and sometimes we don't know them. Interestingly, China, they have introduced functional foods in their national guidelines, which is adopted for COVID treatment, or we can say the therapeutic guidelines for the COVID treatment. But it's very late, but for World Health Organization, in last week, they have publicly said they are trying as well to use the alternative medicine or traditional medicine sources to incorporate in the health guidelines how those could be useful for treating the COVID patients or corona patients. So we are suggesting some of the functional foods, as we said, that turmeric, which contain curcumin in high level, all the citrus fruits, ginger, garlics, black seeds, then all the citrus fruits, some other probiotics, some prebiotic, nuts, grape juice, then leafy green vegetables, all this. And some other fruits as well is still to be explored. I think this could be very much effective source of functional food to mitigate the viruses which are very commonly infecting our respiratory tracts. So important to mitigate the debates and arguments, the diversified approach of clinicians. Sometimes the therapeutic approaches of the people who are using the synthetic drugs, they don't accept it. So we need to mitigate this and we need to harmonize this. Then sometimes lack of evidence-based knowledge. This is also very important. We have to focus the evidence-based knowledge on the functional foods. Then our social wrong perception about the intaking of the functional foods. Then our future recommendations for more preclinical and clinical research on functional foods. Then we have to enhance the evidence-based therapeutic approaches for this of functional foods, and obviously we have to ensure the toxicity level, the right doses and administration of these functional foods. If we can manage that and we can ensure all this, in that case, we can think that for next decades, the functional foods would be the choice of our drugs to treat this sort of respiratory tract virus. Thank you so much. Thanks everybody for your patience here. Thank you very much.
Thank you so much, Dr. Rahman, for your very informative speech. You have question from your first question is from Professor Abhishek Das. The question is, can these functional foods inhibit multi-drug resistant bacteria?
True. Very important. Very nice question. Yes. Some of the functional foods, they are evident that they have the capacity. But first of all, we have to know that how the functional foods could be target specific. It depends whether the functional food is enriched with flavonoids or enriched with terpenoids or enriched with terpenes or enriched with steroids. So for the flavonoid enriched functional foods are highly prospective for the antibacterial infections. More important that these functional foods sometimes they are even better than synthetic drugs because of their synergistic effects. As you know that the synthetic drugs, they do work in a single way pathway or single compartmental model. whereas the functional foods, they can do the same work synergistically. I mean, we are using the functional foods in the raw form. We are not collecting or we are not extracting a single compound from the functional foods. Therefore, a single source of functional food might have this multiple types of compounds. So altogether, the beneficial effects and the harmful effects of the compounds existed in the functional food will be harmonized. That can ensure the infection of bacteria could be mitigated. However, still, I think this field is less nurtured. This is less nurtured field. So we have to do more and more research, and we have to nurture this world more. In that case, we can find out the specific field or specific target foods or specific functional foods which are truly functioning against the multi-drug resistance bacterial infections. Thank you so much.
You have next question from Mrs. Sudha Mahindra. Can these functional foods be called as biofortified foods?
True. To some extent, this is true. But biofortified foods, for example, it's 5,000 longs back. In our subcontinent, we use Ayurvedic drugs. That has been updated. The Arabians and Chinese and some other countries, they used Unani foods, Unani drugs. Then we came to herbal drugs. Then we started using complementary alternative drugs. Then we are now saying evidence-based complementary alternative drugs. Currently, we are observing that many of the traditional practitioners, they use the natural resources in conjunction with some of the APIs, active principles, active ingredients. So in that case, the biofortified foods might have this sort of conjunction. However, once we are saying the functional foods, this should be very much free of this sort of APIs. Am I clear?
Yes, yes, sir.
Thank you so much.
Thank you so much, Dr. Rahman. Food is a very prime thing with our life, and your talk was very informative and very suggestive. So it was very enriching for all of us. Thank you so much. With this, we have arrived to our last session.
Can I leave, please?
Yes, sir. Yes.
Yeah, thank you so much. Thanks once again to invite me.
Sure, sir.
Yeah,
Our pleasure.
Yeah, thank you so much.
Thank you. With this, we are going to start our last session. We are honored with the presence with Dr. Sagar Lahiri. Dr. Lahiri has done his MSc from Department of Microbiology, Vidhannagar Government College under University of Calcutta. He has done his PhD in Structural Biology and Bioinformatics Division from Indian Institute of Chemical Biology, Kolkata. Dr. Lahiri has plenty of research publications. He is currently working as a senior research associate in Chemical Engineering and Applied Chemistry Department of University of Toronto, Canada. The topic of his presentation today is how to get into scientific career and how to be successful in it. What I learned from my journey so far is very relevant for all the students now are studying under UG. It will be very informative session for all of you. Over to Dr. Lahiri.
Thank you, Shudhanuka. Can you guys hear me? I'm sorry, can you guys hear me?
Yeah.
And also, can you see the screen?
Okay, I'm guessing you guys can see the screen. So hi everyone, my name is Dr. Shagor Lahiri. I work at University of Toronto. And first of all, I'd like to thank Shutunuka, who's like a longtime friend of mine. We did our masters together almost like 13 years ago. And I'm really grateful to her that she still remembers me and invited me for this speech. And I'd like to thank all the faculty members, the microbiology department, and the teacher in charge of St. Paul's College for inviting me for this speech. So just to give like a little bit broader introduction to myself, I work in Biozone, which is University of Toronto Center for Applied Bioscience and Bioengineering, and my lab it's called LMSC. I have also added my email here, so it's my name, sagarlahiri@gmail.com. So if you guys have any other questions, you can always email me directly. So just to talk a little bit more about my lab is called LMEC, or Laboratory for Metabolic Systems Engineering. So this is my boss. His name is Professor Radhakrishna Mahadevan, and he has a huge group. He has almost like 30 people. So obviously he can't talk to all of them and manage all of them at the same time. So he has me and 4 other research associates. We all lead like 4 or 5 other people with us. So we have a postdoc, a PhD, a master student, and a lab tech who work with us. And the work that we do, you can find out more if you go to this website, which is called lmse.toronto.ca. What I do is metabolic systems engineering. So what is that? I'll just give a brief introduction to it. It's like we decide on a specific end product, a compound that we want to produce, and then we decide on a specific host, say suppose yeast, Saccharomyces cerevisiae. Now, naturally, maybe the yeast doesn't produce this product at all. So we basically find out an enzyme in nature which can produce this compound. We get that enzyme from some other source. Or if there isn't any enzyme in nature which can do that, we basically synthesize a primary protein ourselves. So we have chemists and physicists who help us with the designs. And then we have computer modelers and simulators like Indrajit, who basically try to design the structure of the protein and everything so that we can have our input. And then we have biochemists and molecular biologists like myself who do the wet lab experiments to validate if that modeling and all this prediction has really worked. So that's what we do in our lab. I'm not going to talk more about my research. I have just added a screenshot of my Google Scholar page. So if you guys want to, you can go to Google Scholar and search my name. You can find all the papers that I have published over 2 years. You can read the papers, and if you guys have any questions, you can always email me. So that being said, I'm going to focus on today's topic, which is what does success mean in science? So as you probably remember, my topic was how to be in a scientific field and how to be successful. So first of all, I'd like to explain what I think success means in science. So first of all, don't go into science for money. There are lots of other ways you can earn money. You can also earn money using science, but don't just go into science to earn a bunch of money. It also doesn't mean fame in the traditional sense. Like scientists are not as famous as like movie stars or like football stars. So if I ask you guys, have any one of you heard the name of Randy Shackman? I'm guessing a lot of you have never heard of him. So if I do a Google search and look for Randy Shackman, you will find out that he has his own Wikipedia page. And if you read this Wikipedia page, you'll find out that apart from being a professor at UC Berkeley and the editor of esteemed scientific journals like PNAS and eLife, he's also the winner of 2013 Nobel Prize on Physiology and Medicine. But he's not as famous as some other people that we know. I know this man myself because he visited my lab in Michigan in 2017, and he's a fascinating human being. If you get a chance to spend 5 minutes with him, if you can have a conversation with him for 5 minutes, no matter who you are, you'll probably learn something from him. So one of the benefits of being in science is the ability to get the chance to be able to interact with people like this who are brilliant. So going back to the points that I had, apart from money and fame, what I think success in science means is a scientist is able to earn a satisfactory living throughout their life using a very specific set of skills. So I know a lot of people back in prehistoric times used to do science just for learn, but today most scientists do science as a profession. So we earn our living by science. So let's say someone can hack into my bank accounts today and take all my money, and somehow they can take my house, my car, all valuables I have. I'll still be fine as long as I have my degrees and I have my knowledge of science. I can get a job, and that job is going to pay me enough that I can maintain my lifestyle. Another ability of a successful scientist is to withstand any temporary setbacks. Now, I'd like to give you a shout out to Indrajit. Indrajit David is a friend of mine. We used to work together at Cathedral Mission in last year since I moved to Toronto. Indrajit probably knows about this. When I met him first, I used to work in a lab in Michigan, and that lab had some funding crisis. So my boss told me like, hey, I love the fact that you work for me, but after 6 months, I won't be able to pay your salary. So you should look for a different lab somewhere else. So I started looking, and my CV was strong enough that I found a different lab in the same university. So you don't have to be the most famous scientist in the world. To be successful in science means you can handle any temporary setbacks in your profession. Another thing I think is a trait of a successful scientist is you are someone that others look up to as a role model. Again, I will give a personal example, and Shutumuka probably also knows him. He's a friend of ours. His name is Dr. Subhashish Parik. He was our classmate. He was my classmate in BSc and our classmate in MSc as well. He was not a brilliant student, but he always had his focus on our career development. So I always looked up to him as a role model. So a successful scientist is someone else's role model. And finally, the final trait of a successful scientist is they never stop learning. I'll tell you the story of a man. His name is David Aminoff, and I have his picture here. So he's a 96 years old professor emeritus of biology chemistry division of University of Michigan. I used to work. So he used to work next to me. He was 96 years old, and he couldn't walk without the help of a walker. He still used to come to the lab almost every day and used to do his own research. And then one day he just asked me, hey, do you guys have those fancy DNA extraction kits? And I was like, do you mean like a plasmid kit? He said, no, I want genomic DNA. I'm like, yeah, we have a genomic DNA extraction kit. So he said, can you teach me that? I'm like, no, let me know. Like, I don't think I'm worthy enough to teach you. I should probably get my boss or my boss's boss to teach you. I was like, no, it's fine. So this 96 years old emeritus professor of University of Michigan, which is one of the best universities in the world, is still willing to learn from someone who is like 50 years younger than him, not even 50, maybe 70 years younger than him. So a successful scientist never stops learning. So these are facts that you should remember. These are things that you have to do to be successful in science. Now, the next question that I have for all of you is, what do you think are the most important factors for success in science? Now, if I asked a random group of people this question, I'm guessing like one of the most common answers would be hard work. Some others might say raw talent. Some others might say like to be lucky. So yeah, maybe these are all important factors. But what I think are the two most important factors to be successful in science is number one, to be aware of the opportunities around yourself, and number two, to prepare yourself to avail those opportunities. I'll tell you another story about my own personal experience. So when I was a master's student, someone told me that there is a research institute in Jadavpur called IIC. You can go there and you can work in a lab for 3 to 6 months, and they will train you how to do research. So I learned that this was an opportunity that was presented to me, and I prepared myself well enough that I contacted some scientists. I made my CV, I read their paper, I talked with them about their research, and then one of them was kind enough to allow me to train him in his lab for 3 months, and his name is Dr. Anil Ghosh, and eventually he was impressed enough with me in those 3 months that after my master's, he offered me an opportunity to do PhD in his lab. So I did my PhD from his lab as well. So it's really important to make sure that you know what are the opportunities around yourself, and you need to prepare yourself to get these opportunities. Now, the next suggestion that I have for all of you guys is always find the next 2 steps. So suppose you are doing BSc microbiology. What are the next 2 steps? I know a lot of people, or who, if you ask them like, what's your next step? They have a plan. Like, say, suppose if it's a BSc microbiology, if you ask them like, hey, what am I going to do after this? He or she will probably say, I'm doing MSc microbiology or genetics. But if you ask them what after that, so often you will see that they won't know what after that. So you need to know about the next 2 steps. So I am just giving an example of BSc microbiology. So after BSc, what are your options? Your options are basically broadly categorized in 2 groups. One is to go for higher studies or to find a job. Now, as you all know, I went for higher studies, so I don't know a lot about finding a job, but I still have some general idea. So I have to all of these. I'm not going to talk about all of these points. I'm going to share this whole PowerPoint with all of you guys, so you can read about them, and if you have questions, you can always email me. But I'm going to just talk about a few brief points which I think are very important. So if you're going for an MSc in microbiology or any other related subject, this is what you need to do during your PSc. And the most important thing you should do is study well. I know it sounds like a cliche, but the only way that you can get into a master's program if you score well in your exam, or if you score well in the admission test. Otherwise, you will not get into this master's program. And finally, another suggestion that I have for you guys is make a list. So suppose you want to do a job in research. Find out all the research institutes of universities who hire a BSc technician. I have talked with many people before, and they say like, oh, I'm trying to find a job. I couldn't get any. I was like, okay, so how many different places have you applied to? And they're like, maybe 15 to 20. Like, so I was like, you are a BSc student and you are trying to look for a job, and you have applied for 20 positions, and you are saying, oh, I can't find a job. After my PhD, and I know some of the other scientists will probably agree with me, after my PhD, to apply for a postdoc, I had to apply to hundreds of labs around the world to get maybe like 10 opportunities. So you need to make a list, and the list should be broad enough, and it should be good enough that you can cover all the places that you can get a chance. And then one other thing that I'm going to talk about is like a lot of students after their PSc have this dilemma whether to go for higher studies or to find a job. Don't ask me what you should do. You should decide it yourself by making a pros versus cons list. So what are the pros if I go for higher studies? What are the cons if I go for finding a job, and vice versa? And then you need to decide it yourself. And the final suggestion that I have for you here is do not regret your decision making. I have seen so many people who have done something like they have done an MBA, or they have worked in a company for 2 years, and then they say like, oh, I should not have done that. I'm regretting this. Please don't be that person. Whatever you decide now is probably the good decision. You need to stick with it and to fight with whatever you have. Now, this is step 1 after BSc. Now, let's look at step 2. Let's say you have done MSc in microbiology. Then what are your options after that? The options are basically saying you can go for higher studies, or you can find a job. And again, I have written down a bunch of pointers here, but I'm not going to talk about all of them. I'm going to talk about a few very important criteria. So if you are looking to get into a PhD program in India or PhD in abroad, one of the most important things apart from your NET, GATE, GRE, TOEFL exams is you need to find a training opportunity for a real research experience. So I was presenting a similar presentation last week to my alma mater, Vijayanagar College, and the students. And some of them had this question asking me like, hey, we know that we need to find research experience, and we found one, but because of this COVID situation, those research labs are partially shut down and they have cancelled our training. What should we do? So I told them like, I have written a real research experience. I have not written a wet lab research experience. The presentation that Indrajit just showed, that's a real research. But for that kind of research, you don't have to go to the lab all the time. You can do a lot of the work from home online. So there are lots of labs everywhere in the world, and especially in India, who do a lot of work in like molecular simulations, computer modeling, and all these kinds of things that you can actually get a training online from your home. And also, the point I'd like to talk about is like if you're applying for a PhD abroad, if you're thinking about it, apart from your GRE or TOEFL, you need to prepare your essay. So people in US and Canada really like the fact how passionate you are about science. So you need to tell them your story, how passionate you are. So I used to work under a scientist named Dr. Mara Duncan in the CDB department of Michigan, and she was also the selection committee member of the PhD committee, the PhD applications. So she singled out an application and showed it to me once. This application came from a guy whose name was Seth, and he had a degree in biology from Wayne State University, but he applied from prison. So when he applied initially, he said like, hey, I have a degree in biology, but then I got involved in the wrong crowd and I ended up in jail, and now I'm in prison. But I realized that I need to get my life back on track. So I'm applying to you guys. I know that I can't start my PhD next year, but please tell me what can I do from inside the prison to improve my chances. So all the professors in that selection committee, they personally sent him a textbook each to his prison cell, and they all contacted prison warden to request him to give this guy access to the library or online access. So after 2 years, in the third year when he applied, he was the first person who got accepted in this program. So the summary of the story is, if this guy can get a PhD opportunity in a place like Michigan, what's your excuse? So please don't hide behind your excuses. Whatever your problems are, you need to find a solution to those, and you need to find a way to move beyond those. Another thing that I wanted to talk to all of you guys today is who gets the job. So I know some of you might start a company after your PSC or your MSc, or you can do a business and you might be successful. So that's great if you do that. But a lot of you will probably do jobs your whole life. And one thing I should mention is compared to the previous generation of our parents, today people apply for jobs their whole life. My boss, Professor Mahadevan, he still applies for jobs. He's a professor, but he's applying for positions like the departmental head or the president of the university or something like that. So one thing you should know is the salary, the experience of different jobs might be different, but the basic selection criteria for any job is basically the same. So who gets the job? It's someone who fulfills all the educational and professional requirements for the job. Now, that doesn't mean that if you have all this, you'll get the job. It means if you don't have this, you don't have the educational professional requirements, you will definitely not get the job. It also means someone who has sufficient communication skills, because you might know everything in the world, but if you can't communicate that to the interviewer, you'll not get the job. Also, you need to have enough technical knowledge to answer expected questions. Whenever you go into an interview, especially in science, as a GRA, project assistant, or research associate, whatever, they will probably ask you a technical question like, hey, do you know what's the difference between protein and DNA, something like that. So you need to be able to answer the technical questions you should not have any obvious red flags. So I don't know if you guys are familiar with the term red flags. It means doing something weird in the interview. So I was interviewing a bunch of candidates for a lab tech position in my group this year. And there was this guy who had a great CV and everything, but then he said something really weird. He said he doesn't like getting orders. He must rather give orders himself. So that was like a big red flag for me, because I'm supposed to give him orders and he's supposed to listen to my orders. So obviously that guy didn't got the job. So don't say or do something which basically makes the interviewer question whether they should hire you. And finally, it's someone who can gain the trust of the interviewer to risk. Now, you might be wondering, what is the interviewer? So I'm going to explain that in the next slide, but before that I have quoted here, which is email the real PI or the real supervisor. You guys will probably apply for jobs, and these days you have all these websites like Indeed and Monster.com and Naukri.com and whatever. And you can apply to those online portals and click, and then application is sent. But I will tell you another story of my personal experience. When I was applying for this job, the current job I am holding, last year from Michigan, I saw this ad in Indeed, and I went to the online portal and did click, and the application was submitted. The next day, I received an email from one of my department's admins from Receptor Toronto saying like, hey, we are sorry, we don't think you are a very good fit for this job, so you are not getting considered for this job. I was like, oh, okay. But another thing that I did on that same day was I found out who is the real supervisor or the scientist whose lab I'm supposed to work. I found out his name is Dr. Mahadevan. So I Googled him, I found out his email, and I emailed him saying like, hey, I saw that you have posted an ad for a research associate, and you have we are looking for these criteria. I have done those things, I have experience in those things, and I think I'll be a good fit for your lab. So if you can consider me, we can definitely talk. So 2 days after I got rejected from the admin, I received an email from Krishna, who is my supervisor right now. He said like, hey, I like your CV, let's talk more. So I was like, yes, and I talked with him, and I got the job. So even after you get rejected by the admins or the HAPP, you need to understand they are qualified in their own way. They know what they're doing. But sometimes, because our subjects are so multidisciplinary, sometimes they might not understand your value. But if you are applying to a company or if you are applying to a research lab, the technical person that you are supposed to report to, make sure that your application reaches them, because they are the only people who can definitely understand your value. Okay. So getting back to what the interviewer is risking. So when Krishna hired me, he was risking his own money. And by his own money, I don't mean like his own money, but his research grant. He hired me and he offered me a salary. So if I don't do my job, he's basically wasting his money. But when I am hiring somebody, I'm risking my own job. When I'm hiring a lab tech, and if they don't do their job, then I'll have to do their job as well, or we'll both get fired. So when I am interviewing somebody these days, what are the determining factors that I look for? And this doesn't matter whether you are applying for a GRF position, a research position, a postdoc position, a lab tech position, whatever your job is. Determining factors are basically the same. The first thing I look for is the absence of red flag. So don't come to an interview wearing like a jeans which has holes in it, or do something weird. Most interviewers are basically very safe kind of people, so we would also go for that kind of person. The next thing I look for is like whether the candidate seems to have enough theoretical knowledge. I can only guess if they have enough theoretical knowledge, but if they can give me that idea, that makes me think they probably know, that I consider them a positive candidate. I also look for candidates who have relevant hands-on experience. And finally, whether the skill set and the personality fit the team. And again, I have another pro tip here, which is don't just say, prove it. Say suppose if you are applying for a job as a GRF in a couple of years, which is the junior research fellow, and you tell your interviewer like, hey, I'm an expert in protein purification. Don't say that. You can say that I worked in Professor X's lab for this many months or this many years. I have learned a lot about protein purification. I have learned about reverse space chromatography and amino acid filtration and all these things. And I really like it. So that basically tells the interviewer that you know about protein purification, but you are saying it in such a way that they believe you. So make sure that whatever you are saying, you are basically proving it with your own words. Because most of the times interviewers are very smart people. They can understand if you are lying or not telling the truth. So another thing that I wanted to talk to you guys about is like, so for all of you who are interested in research, one thing you should know is like the difference between a job in a company and research is like, there are companies like TCAs and CTAs who go to like colleges and campuses. So you don't have to find the company. The company finds you. But if you are trying to do research, you will have to find the lab. So don't just go for the first lab who accepts you. Go for a good lab, because if you are stuck in a bad lab, then your career is basically ruined. So how do you find a good lab? And I can show you the example of my own PI. So the first thing you should do is Google the PI. So let's say the name of my professor is Radhakrishnan Mahadev. I Googled him. He's the first website that came in Google. And so this is University of Toronto's website. So you go to this website and you can click on this, which is his lab website, which is basically this website that I showed the pictures of. And you can find out about the research in that lab, what kind of papers they have. And you can also go to a site, as I mentioned earlier, Google Scholar, and you can look for Radhakrishnan Mahadevan and what was the name of your PI that I'm looking for. And you can find out what are the papers that this guy has produced in, say, this year or last year. And you can read some of these papers. You can try to understand what kind of work they're doing. And then if, say, another thing you should look for is like the alumni. So if you go to our lab website, there's probably somewhere that they have an alumnus page. So yeah, if you go here, you see the alumni. So it basically says who are the graduate students and what are they doing now. So when I was applying for Krishna's lab, I found out this guy Jonas and this guy Shivas. So I realized that Shivas is still in Canada and he was in this company called Sanofi Pasteur. So I emailed him and I said, like, hey, I was offered a position in the Mahadevan's lab. I realized that I have worked there for a couple of years or 3 years. What do you think about this lab? And this is my CV, and do you think I'll be a good fit for the lab? So he said, like, yeah, it's a great lab. Krishna is a great PI. You should definitely work in it. So you can look up the alumni. Say, suppose if you are interested in a lab in IICD, which is Krishna Chattopadhyay's lab, you will find out me as one of the alumni. So you can probably email me and say like, hey, I wanted to know if Dr. Krishna Chattopadhyay's lab is a good lab to do my PhD. What do you think of it? So if you email people like that, they will respond to you. And another thing you should do is you can email the PI and the seniors in the lab asking about information about their research. Don't email people and ask for a job or like a training opportunity. We receive those every day and nobody likes them. But if you email a PI, like a scientist, or principal investigator, or some of the seniors in the lab, asking like, hey, what kind of research are you guys doing? I'm telling you, 90% of the researchers will be really happy and they will reply back to you explaining what they are doing, because scientists love talking about their science. And also you can ask them if they're willing to talk to you about their research for a day. I remember this girl, Mehi, from my PhD lab. When I was in my third year, Mehi was like applying for the PhD in my lab in IIC. So she was applying from Rajasthan and she said like, hey, I basically do a master's in a university from Pakistan. And I don't really have a really great experience of research, but I was just wondering, would you guys be willing to just talk to me about your research for a day? I can go to Kolkata. So both me and my boss, we are very impressed that this girl is willing to travel from Rajasthan to Kolkata just come to our lab for one day to learn about our science. So these are the kind of things that you can do to impress a scientist. And especially if that lab is in Kolkata, it's not much of a big deal. Like, you know, right now, because of this COVID situation, they'll probably not let you come to the lab, but you can probably talk to them online. You can have a Skype or like Google Meet or something, and you can ask them about their research, and they'll be happy to discuss with you. And try to learn from them on day one, get some paper recommendations, and read those and try to understand those. So I understand if you are BSc students or MSc students, you might not understand a paper from my lab or say Indrajit's lab or some other labs. But if you can show a scientist or a PhD student or a postdoc that you have tried hard enough to understand, that's good enough. So if you just try to discuss with them, that's all you have to do. Now you just let them do the talking. So I did everything from step 1 to up to this step in my lab doing my master's with a guy called Orgoda, who was my senior. And when I did all of this, then after a few months, Orgoda said, like, you know what, you should do PhD in our lab. I'm going to help you. So if you do all these things well with a number of different labs, at least one of them is going to help you to do your PhD or whatever else you want to do in your life. So I have a few suggestions about what to do about after you start your PhD. I'm guessing most of you are not there yet, so I'm not going to talk about all of this. But a few things I will just point out is valid from now, like wherever you are. So one of the things that most PhD students do is they read the chapters related to their project from their master's or their PhD textbooks. So please try to read your textbooks well now, so that when you start your PhD, and if you're reading your textbooks again, it will be easier for you. Another thing that I'd like to mention is make friends with others who are trying to do better research. And it doesn't only mean better research, but trying to improve themselves. Because your friends today are just your friends today. But in 5-10 years, they will be your collaborators as well. So just last week, I needed a GFP classmate. So GFP, green fluorescent protein, is a dye or like a tool basically we use in our molecular biology exams. I did not have like an alternative to GFP, but I knew a friend of mine who Indrajit also knows. He works in Michigan, and he had a different alternative to this protein. And I called him and I was like, hey, you have this M scarlet, right? Can you send me that plasmid? And he did send me that plasmid on that same day, and he did overnight shipping. So I received the plasmid the next day. So your friends are just your friends that you hang out with today, but they will be your collaborators in like 10, 15 years. Also, another thing you should start doing from now is how to read a scientific article. When I was in my master's, it used to probably take me like a couple of days to finish a paper, a scientific article. Now it probably takes me a couple of hours. And I know for Krishna or Ryan or Mara, who are my previous supervisor, it probably takes them like 10-15 minutes to read a paper. So this is something that you should also try to learn. You can just Google how to learn how to read a scientific article. You can search in YouTube. There are probably a bunch of TED Talks which will explain to you. Another thing that I would like to mention here is make a weekly calendar. Like, what are the subjects I going to study this week? And then after the week is over, try to look back at your calendar. Like, have you done all those things? So I'm just continuing with the tips is like back calculate your timeline. So this is for a PhD student, but even for a PhD student or MSc student, it's the same. Let's say you have an exam in next March. So you have this many months and you have this many different subjects, and each subject has this many different chapters. You need to back calculate your timeline. Like, if I want to keep 20 days for the revision of the whole syllabus, how much time do I have to spend now to read through my whole syllabus? And another suggestion that I have here is what Ms. Chengu was talking about, is solve problems in your personal life. Every time you have a problem in your personal life, you will take a hit in your professional life, in your productivity. We all go through a lot of different transitions during our MSc and our PhD, whether it's getting married, having children and all these things. But whenever you have a problem in your personal life, you should know that it's going to affect your professional life as well. So try to solve your problems in your personal life. And finally, I'll just show a card here. So this is like my chart of time versus knowledge versus productivity. So as Mr. Shimbutu was talking about, is like the ability to learn and everything. So this is 10 years time frame. So say suppose you have started at like first year of your PhD, you have finished your PhD on 5th year, and then 5 years after that. So these bars are learning or knowledge, and this orange line is your productivity. So you can probably say like this is my card. So on the first year of my PhD, I knew nothing and I could get nothing done. From my second or third year, I was learning a lot, but I did not saw that much increase in my productivity. But after a certain time, like maybe like the 4th or 5th year of my PhD, when I learned enough, then I saw a certain spike in my productivity. It's not only papers that I published, it's also basically the scientific experiments that used to take months to be successful before started working on the first attempt. So in your PhD, there will be a time when you are learning and you're not productive enough, but you should keep on learning, because then they will see a spike like this. And then after my PhD, I moved to a different lab for my postdoc, and I was learning more again. But then my productivity was not that high again, because I was trying to learn enough to do a successful research in a lab like in University of Michigan. And then after maybe 3 years or something, I saw again a spike in my productivity. And today, I'm probably here, I'm more productive than ever before. And I can get more experiments done in a week or maybe a month now than I could do in a year, a full year in my PhD, first year, second year. So yeah, so that's all I had to talk to you guys about. I hope it is all helpful. And we have a dinosaur who say thanks to all of you, and he's asking for questions. So don't think any question can be silly or stupid. Like I'm especially asking students to ask me any questions that you might have. So yeah, fire away.
I must say, very, very delightful speech, Shagar. As you said, you have passed out 13 years ago, but still most of the undergraduate students have the same question: what can I do after completing the graduation or post-graduation? So your speech is very relevant and helpful for all the students. And during your session, I was getting so many messages. All were saying that we should keep your session at the first of the today's session. But for your favorable time, we had to keep it at the last of our session. But all of us really enjoyed your session. And you have questions from Shubham Nondi. The question is GRE necessary for PhD in foreign country?
So I'll tell you, like the answer was yes a year ago, but it's different now. A lot of universities in both US and Canada, they try to be better human beings. And they have understood this year that it's really, really difficult for people to study for GRE or to go to a center for GRE, and it might be risking their health. So a lot of the universities are considering waiving the need of GRE. So I know like MD Anderson last month declared that they don't need a GRE. And one of the most important things that I have always seen that people care about here is if you have some research experience. I have like the most students that who are doing PhD in our lab and in my previous labs, they had probably worked as a project assistant or something similar in an Indian research lab for like a year or 2 years, something like that. So this is something that most universities really, really give top priority to.
Your next question is from Rochona Pramani. She was saying, I am currently pursuing masters. How can I approach a research lab for a hands-on training in Kolkata?
It's very simple. So Shudhanuga knows these people. So me and my friends, a few of my friends, we made a list of 50 labs that we can approach in Kolkata. So we went to the websites of, say, suppose IICD, Post Institute, Indian Association of Cultivation of Science, Rajabad Science College, Paligon Science College, and then there was a bunch of other institutes like, and today there's probably more. So you can go to all these institutes' websites and you can find out all the different labs, and you can choose. Basically, this is like the fun part. You can choose any number of labs that you want to, and you find out the email addresses of the main scientists or some of the senior students. And don't ask them initially that I want to do a training in your lab. They will be pissed off and they will not reply to you. So start by asking about their research. If you can read some of their papers and you can say like, hey sir, I have read some of your papers. It's a really great field that you're working on and I want to know more. Can you please suggest a few more relevant papers, something like that? You need to start a dialogue with them, and when they are okay with you, when they think that you are genuinely interested in their science and not just a research training, then you can ask them and they'll probably say yes.
Your next question is, that is from our faculty Orpita Komkar. What should be the simplest route if getting a doctorate degree in science instead of philosophy? Or how to obtain DSc degree in India?
I don't know, basically. So one thing I have realized is like when I applied for my PhD, I don't know the difference between DSc and PhD. Most of the people that I know who are in science in IICD or in University of Michigan or University of Toronto, most of them have PhDs. I'm guessing that maybe DSCs are somehow more important, but in most cases, a PhD degree is good enough.
Thank you, Sagar. Thank you so much for your
I'm guessing these days, most probably, a DSc is more like an prestigious kind of a degree that is given to some selected scientists. Like I know University of Michigan gives like honorary DSc degrees to people who have worked in science for more than 30 years but never had a degree or something.
Thank you so much, Sagar, for your presence and for your wonderful talk. With this, we have come to the end of today's session. We have our college secretary with us. I would like to request her once again to share a few lines of him for this seminar.
I've got nothing more to say than that we have had an excellent evening of presentations and we have learned such a bit from each one of them on such varied subjects. Thanks to each one of them whom you have invited, and we are so grateful to them. Thank you so much.
Thank you, sir. Thank you so much. Now I would like to invite...
One thing I would like to add is like if you guys can give me an email address, I can send my PowerPoint and you can share it with all the students.
Thank you so much. Thank you. That will be very helpful. Now, I would like to invite our faculty, Abhishek Dash, to address a vote of thanks.
Thank you, Shudanuga. Our honorable speaker, my respected teachers, and all the participants across the India and abroad. It's my privilege to have been asked to propose a vote of thanks on this occasion. An event like this cannot happen overnight. The wheels start rolling months ago. It requires planning and a bird's eye for details. We have been fortunate enough to be backed by a team of very motivated and dedicated colleagues of our St. Paul's family who know their job and results are oriented. On behalf of the Microbiology Department, St. Paul's Cathedral Mission College, I wanted to extend a very hearty vote of thanks to all the participants who blessed us with their presence and took out valuable time from their busy schedule. I would like to extend a special thanks to the respected speakers of this event, Mr. Saurabh Sengupta, Professor Atiyar Rahman, Dr. Indrajit David, and Dr. Sagar Lahiri, for inspiring all of us with their words. I must mention our deep sense of appreciation for our governing body secretary, Dr. G.R.B. Alfred, for his encouragement and support. Thank you, sir. I am immensely thankful to our respected teacher in charge, Professor Devashish Mondal, for joining us today. I must mention our deep sense of appreciation for our bursar also, Professor Shubhendu Kanjilal, for his encouragement and support. We have been fortunate to be assisted by our IQAC coordinator, Dr. Shobboshachi Mahapatro. Thank you, sir, for being with us. Finally, I would like to take this opportunity to place on record our hearty thanks to Antariksh Venkataraman and his team from our microbiology department for the perfect technical support they extended for this webinar. Once again, I want to state that we are grateful to all these speakers. We thank you for being with us in this evening. It's been a great pleasure. Thank you very much.
Thank you, everyone. Thank you for your valuable presence.